Ok. Bueno, pues quiero iniciar agradeciendo a todos los asistentes a nuestro último día en este primer congreso. Y bueno, vamos a iniciar presentando a nuestro primer ponente, el doctor Pierre, Pierre Henry. Este doctor, además de ser un miembro honorario de nuestro grupo de investigación en Landsbiodit, es investigador del Instituto Nacional de Investigación en Salud y Medicina de Francia. Su amplia expertise se ve reflejada en un elevado número de publicaciones donde aborda temas como es el reconocimiento, la adhesión y la mecanobiología celular. Es especialista en el desarrollo de técnicas que permiten medir la fuerza tanto de partículas como de células individuales, empleando microscopía de fuerza atómica y pruebas de fuerza de membrana. Además de todo esto, es responsable de nuestra colaboración con Francia en el proyecto internacional Biophysique Inmuno. Le agradezco mucho su asistencia a nuestro congreso y bueno.
Muchas gracias. Y muchas gracias a todas y todos para estar aquí. Voy a hacer un switch in English because it's easier for me to speak in English for science, I would say. So for the ones who were there last year, it will sound a bit reminiscent. Okay, the sound went off. Oh, no, it's fine. So here I will try to guide you today around T-cell mechanotransduction across scales. You've been hearing during the week a lot about these several scales, several tools. So you will see things that will remind you, and so I will shortcut sometimes through the presentation. Here you have the very well-known Diego Rivera mural that you have in the Palacio de Bellas Artes. And what you will see here, look at this small corner. So this man is controlling nature, and in the corner you have a small microscope and you have something, but in fact, if you zoom in, is reminiscent of either an eosinophile or a neutrophile. Okay, so that's already my starting point, but I will focus on lymphocytes. As for the talk of Jay Gross of this, the beginning of the week, and the talk of Laurent of yesterday, this is the same kind of cells. So I will simply summarize very briefly. First, the role and action. Again, the same video that we've seen during the talk of Jay. A T-cell scan an antigen-presenting cell. Antigen-presenting cell could be a physiological one, like dendritic cell, macrophages, others, B cells, and so, or it could be a cancer cell. It has to present a message that the T-cell will read by scanning it very actively. In fact, it touches, pushes, try to read the surface as you would do if you were blind, basically. So it has to read the antigen and then react. And these cells function through their T-cell receptor that Laurent presented yesterday. You have different classes with different roles. Some, the CD4 plus, are made for calling partners to build up the inflammation process. The CD8 plus are much more reminiscent of what Laurent presented yesterday, the NK cell. They have to kill the other one. These are just cells that have totally, and I would say globally, similar processes, but they have one co-receptor that is different, the CD4, CD8. All in all, Jay and Laurent and also Julien presented the fact that TCR-PMHC has to be fast, reliable, and sensitive to lead to a proper immune reaction. And the T-cell, in fact, again, just to emphasize this fact, understands classical molecular scales and classical molecular cues, the TCR, the adhesion molecules, the co-receptors, and everything. This, the T-cell can organize. Here is the T-cell at the bottom, the antigen-presenting cell on top. The T-cell can organize the molecules, forming clusters and, in fine, immune synapses. These are very highly organized structure where you have the sorting of the molecule depending on the size, but also depending on their function. So if I say sizes, you can directly see that some physical parameter at molecular scale can be important for those molecules to function. But not only that, but over the last decades, it has been more and more recognized that those cells can recognize the influence of forces. And these forces, as Laurent mentioned yesterday, at cellular forces, and in the lab, we've shown that TCR molecules feel forces. It is also what you've seen during some talks this week. TCR PMHE can be a catch bond. This is what Jay and Laurent also presented during that talk. And what you've seen with Julien's talk is that they understand also substrate mechanics or large-scale mechanics. They can adapt to the substrate they are facing. What is very interesting is that the forces are also used for the T cell to perform their action. Because if they need to pierce one of a cell, for example, using the granzyme production, it's easier to pierce something when the thing is under tension. And this has been shown that the T cell, in fact, put the other cell under tension before performing their action. What is more interesting for me is that the partner cells, in particular the immune cells, dendritic B or macrophages, they have different mechanical properties. So this is in fact possibly another signal that the immune partner cell gives to the T cell, and moreover, they are able to modulate these properties as a function of where you are in the inflammatory process. If you take a DC dendritic cell that is not primed, it's kind of soft. If you prime it, that means that it has something to say, it becomes stiff. So this you could think directly. I could have the message from the molecules and at the same time, a material-like message for me. So usually people perform the experiments either on artificial substrate with one cell line, with one molecule, only looking at forces, only looking at signaling. So what is missing in all the story and what we try to do in the lab is to mix up different techniques and different approaches to mix up, to get a more global view of what's happening. And this is typically what we've been doing collectively, just to present you a bit the work of our lab, of adhesion inflammation lab in Marseille, but also our collaborators. We've been starting from single molecule, looking at, for example, the influence of the fact that the molecule can diffuse in an artificial membrane, the role of this lateral motion in the organization and response of the cell. This is the work of Laurent Evgelia. The way, if now, instead of putting an homogeneous substrate, you present discrete spots, all this structure that reminds you of what is present on a dendritic cell. What is the importance of this lateral separation? This is also Laurent Evgelia. The way the cells, for example, can spread on mechanically different substrate, what is happening if you mix this with the presentation as a small bouquet of molecules using this transfer nanopatterning. If you shape them to control the tension, for example, how this play onto their mechanics, plenty of different stuff. So today I will try to guide you through different scales, through different techniques, and try to show you how we intend to look at the same system from different perspectives, with different tools, with different scales, and try to bridge as much as possible the scales and the approaches. So I will start with single molecule, then go on single cells, then go on what is the link between the recognition molecules and the cytoskeleton, with different ways of looking at it, and trying to bridge the scales between the molecular recognition forces and the cellular forces. So to start with, I will just remind you briefly what is atomic force microscopy, even if we had a very extended talk about it. It's a microscopy that measures forces and uses forces to see objects, a little bit like if you were blind. If you are blind, you touch, you can feel the substrate. This gives you the roughness. This is what we've seen during the previous talk on AFM. But the AFM I'm using, like the way I'm using it, is much more like pressing on the substrate. If you touch something like this, you can see whether it's stiff or soft. And pulling, you can see whether it sticks or not. So that's the way I'm going to do. So you have a spring that is called a cantilever, but it's detected by a laser that bounces the laser on the diode. If you are far, nothing happens. If you press, you bend, so you can detect the contact forces. If you pull and if you stay stuck, you stay in contact and then you jump out. That gives you curves like that, where this part gives you the mechanics and this small jumps gives you the adhesion, basically. So we first went for single molecule measurements because there was a huge question is what is the influence of forces on TCR-PMHC recognition? So you can see the date of the paper, not on this one, but it's 2011. So these experiments are back from 2009, 2008, I would say. So it's not yesterday. So the question we try to ask is, if you change the peptide that is presented by the MHC molecules, is the force that the TCR will be able to exert on it different? You know, the T cell is in fact scrolling around the cell, pulling on different molecules. Is it a way to distinguish between a good, let's say good, you don't have to act, bad, you have to do something against the APC? Is the force a good predictor of the type of peptide? So to do so very naively, we went on putting peptide MHC onto the cantilever, use non-activated, very gently adhered T cell, and we probed, but with very dilute molecule, as Laurent mentioned yesterday for flow chamber. This is a condition that we need to be able to be sure that we look at single molecule events. So very often, nothing. Eventually, you will detect small jumps, and you see that the jumps are 20 piconewton, that means on the same range as the noise. So it's already something that tells you that this recognition is very on very low forces. The usual forces for molecular bridge is much more on the order of 100 piconewton. At those scales, a factor of 5 is enormous. The Brownian motion is on the order of 4 piconewton. So you really are on the verge of all the technical limitations. And what we've seen was at the same time very positive and very depressing. So we've seen that varying the peptide and varying the presence of co-receptors, we had purely specific interactions diluted in all these single molecule measurements. So this is 100,000 forces scales for this kind of stuff. What was also very good, that we've seen that multiple bonds can occur. And you had more bonds, more multiple bonds, when you had the peptide that is recognized as aggressive, but also when you have the co-receptor. So this is coherent with the bio. But what was very disappointing is after 4 years, the rupture force appeared to be non-sensitive to the peptide. And then the question was why? So forces is not a good predictor. 5 years later, people have looked at the TCR with a technique that had 2 piconewton to 5 piconewton resolution. This is what is called biomembrane force probe. Julien talked about it yesterday. And what was the point is that it was not the force, but the duration of the bond under a given level of force. What is the subtlety? Is that with the same technique, we had too much noise because the max duration occurs around 10 piconewton. A factor of 2 killed our effect. So we were not able to switch. The second one, we are not pulling the right way. When you pull like this until rupture. Or you can pull, wait, under a given level of force. The first one was too violent for the bond, and we were not in a good range of forces. So you can see that this is just for underlining that you could do an experiment during 4 years, have thousands of data points, get something that looks coherent, but then face the problem that you are just a side of the good way of measuring the stuff. So AFM was not a good technique for that, but since we had it, we said, okay, let's use it in another way. When I touch something, I detect the position where the object is, okay? When I want to present a cell to another one, I want to be able to say, this is when the cells have been touching. So what is the good point in that stuff? I could use the AFM as a hand, grab a cell, put it somewhere, and detect the force. When I have forces, contact is made. And we went for that. We tried to make, some standardization of looking at forces and looking at T cells under forces using a model system that we can tune at will by transfection. So I won't go into the molecular details of that stuff. It's not important at that point. And our goal will be to put this antigen presenting cell glued on an AFM. This is our hand. And put a target cell on the other side. The problem here is that you have cells that are unique. So every force curve will be depending on the cell. So the idea was to try to look at the ways to characterize and to standardize those cells. If we were to press on the cell and see the action, we need to record a reporter. The reporter here, the most trivial one, is calcium fluxes that you've seen with Julien yesterday. So the idea was to have the cells here adhering but not activated, loaded with calcium probe, and then we generate a signal by bringing the other cell on top. First difficulty. Second difficulty, when you put cells on substrate, you all know that they will have different shapes. As I mentioned, their tension could be also another problem. So if you put them a lot, or if you don't put them a lot, then you will have different starting mechanical conditions. So how do you shape a cell and how you constrain the cell? So we went for shaping the cell, test that we can activate them and check the early activation of them by traditional means, fluorescence microscopy, then do the same on the forces using an AFM, and then use this AFM to set the zero interaction contact time. The first difficulty for these cells is that they are super reactive. So whenever you do an experiment, whenever you shear them, whenever you plate them, whenever you do something, they can react, they can activate, and once they've been activated, they get energized. That means that they are no more able to perform any action. So the first thing was to find a good molecule to adhere them. If you use this NDCD3 that we heard about many times by Julien, for example, this is an activating molecule. So you put the cells on it. Here, one line is one cell. The color, the more red it goes, the more calcium it expresses. You put it on NTCD3, you see them, all the cells almost are flashing. Activating condition, adhering, but activating condition. On BSA, no adhesion, very little activation, but coherent. Anti-CD45, CD45 is a very long molecule with sugar residues that is very present on the immune cells. It has a role also, the activation, but these are subtleties. What you see is that when they adhere, there's no calcium fluxes. And if you record, I don't go into this stuff, but basically we recorded the position, the fact that they are activate, and also the speed if they move. With anti-CD45, they are not moving. So it's a good way to glue them, not to activate them. Here it's a good way to protect the surface for the adhesion. So the goal is now, if you have this, can you reactivate them? You put soluble anti-CD3 and you get the same kind of fluxes after the cell have been adhered onto the substrate. So we have a good way to have innocent cells adhered on the substrate. And to shape them, we went for micropatterning. You will hear maybe more about this kind of patterning techniques, but we already seen some during the talk via the talk of Amarani yesterday. But here, we instead of printing the positive part, we chose to print the negative part. That means the repulsion. This is here what you see. This is the BSA, a biotinylated BSA labeled with fluorescence streptavidin. Why? Because our antibodies were sensitive to the desiccation that you have when you stamp. There's a slight step where you have to remove the excess of ink, and that was killing the molecules. Also, backfilling with the antibody is better because you control better the amount of molecules you put in the stuff. So you can play on the dose very easily. So you have here the spots, here you have the cells. Here, this is interference microscopy that Laurent presented yesterday. You can see that the cells are adhering round, and they are very round shape. This is confocal. This is a Z stack of confocal. Basically, what you have is this BSA protected surface, CD45 zones, cells adhering on it after a couple of hours. So now you can say, are they homogeneous in mechanics? Are they able to respond? The first stuff, you use AFM. So you have your cells here. You have an AFM that we decorated with a bead to be gentle. Again, this is a technique of Dieter. We press on them, we use a model, the Hertz model, very classical thing, and we extract the Young modulus. The higher the Young modulus, the stiffer the cell. That's the stuff. And what we've seen here in black, at 25 degrees Celsius, we were extending the size of the pattern, 10, 15, 20, and then let the cell free. What we've seen is that the Young modulus is increasing when we increase the size of the stuff. Yesterday, we heard by Julien, but in fact, measuring this young modulus was a BS way to access the tension. The more tense, the more young modulus. So this is reasonable. But at 37 degrees, we lose the tendency on the patterns. Why? Because the cell has to adapt, and the cell in fact adapts. First, drawback. We pattern them, but we don't tune the mechanics at 37 degrees. Small trick. Second trick, we load them with calcium. And we measure the mechanics. What happens is that when you load it, simply loading them with the dye kills the effect on mechanics. Again, why? Because the dye has to enter. Since the dye has to enter, you need some surfactant or you need some molecules that will modify the membrane and potentially the cytoskeleton. So this is what kills, in fact, the tension. So if you get this, very disappointing again. You can pattern them, but you don't control the mechanics. If you load them with a calcium dye, you can load them. It's very homogeneous, but you kill the mechanics. So what can you do with that? First, you check what I've said before, but if you seed them and if you have them shaped, you can reactivate them. It's not deactivating them. So it's not an effect of energy, what we've seen at 37 degrees. You can, by chunking some anti-CD3 solution, have a very good, very good reactivation of the cells on continuous and on pattern. And if you throw on them some cells that are presenting the right peptide, they do the same kind of stuff. Here it's on pattern. You can see that the reactivation is similar between continuous and pattern. Everything is similar. So to solve this and to make it short, we had to build a new system based on a molecule that is not present in the immune system, to have it expressed by T cells, to have an independent adhering molecule from any kind of stuff. In fact, that can respect the fact that we can control the adhesion. But this is another story. So what did we do with that? Again, you fail, you were one step back. And one step back is simply to try to build this system without controlling the shape, to say it could be already enough to see something. So what we did, we rendered the cantilever adherent by putting a lectin. This is just micropipette. Again, you recognize what Julien had been presenting yesterday. We used it just to check whether gluing is possible and whether the gluing is strong enough. What we've seen is you can glue a cell in a second, and then you can pull like hell, the cell won't detach. So all the unbinding will be here. And then once you have your cell, in fact, you simply fish at it like this. You have a sticky cantilever, you find your cell, touch it, lift it, and then you have an object. And then you can bring it on another one, and then you can press. This is here. You can press, you can make this contact time, and then you can separate them. But if you want to follow the calcium, you need to have a timer between the AFM and the fluorescence. And here, you're lucky. You're lucky, why? Because whenever you put on a fluorescent light, whenever you take a picture, the cantilever that has gold on one side and no gold on the other side reacts as a thermometer. In fact, it feels the stuff and jumps. So you have an artifact that, in fact, that you can use to know when, in your force curve, you have connection. You took an image. So you can realign using this artifact, these small spikes that are very regular, very rectangular. It depends on the type of cantilever, it depends on the light, it depends on the power. Okay? But you can use this timer to align two microscopes with no cables, no electronics, nothing. So it's the lazy man approach. So what is very interesting is that if you do that, and if you activate the cell, you can indeed record the pattern, force of pattern, and the calcium inside. And here you can see that when calcium rises, cells start to do something and defend against the pressure. So this is coherent with what you want to do. But what is interesting also, long story made short, you can use it reverse. This timer, if you activate the cytoskeleton, you can know when you started to activate it. And for example, here you're just pressing like this, many times. And at what time you flash and you activate the cell, you can record when and see the modulation of mechanics. At the moment, we are using this with Julien to try to see with AFM the influence of the modulation of the mechanics when you have a fluorescent dye. The stuff that is presented at the end of this talk yesterday. about the ROS molecule that could be expressed. Going back to TCR-PMHC, this is what you get. Fluorescently loaded T cells. Here, a COS-presenting cell, an antigen-presenting cell on the cantilever. This is the one we test. This is a control that is just next by. The control, no fluorescence. When you press, after roughly a minute, you get calcium fluxes, which is typical for the stuff. So we have a good tool, but it's very delicate to put in place and very long to use. So here, the signaling and the calcium, in fact, complexifies the situation because you have feedback loops between calcium, cytoskeleton, activation, adhesion, mechanics, and all this stuff. So we gave a try, and Julien presented that yesterday. He had some evidence that the mechanic of the T cell is modulated very early when there is some recognition. Yesterday, you've seen this is the data that he presented yesterday. You touch a T cell with a bead, and immediately the Young modulus increases, the viscosity increases, and so on. So what we went for, we started to see also in some of our experiments that T cell versus B cell, that was the question of yesterday, were discussing mechanically. One was throwing something, the other one was responding, throwing something, responding, throwing. So we were saying, okay, maybe we can build a system to test this with this kind of presentation. And using AFM, in fact, we killed the signaling. We removed all the sarkinase signaling. So we went just for what is very proximal by using PP2. And then we did present the T cell, get a contact time of 1 minute from what we've seen before, and just let the stuff relax. First, we checked that all the contacts between the different conditions with peptide, no peptide, different peptides, and so were insensitive to the peptide, which is logical. 11 amino acids should not change the mechanics of the contact. Then we tested the adhesion, and the adhesion was sensitive to the peptide. So it was a specific recognition of what's going on. But really surprisingly, and in fact, totally coherent with what Julien showed, the relaxation time during the 1 minute, the T cell was recognizing that there is a peptide and modulating its relaxation properties, being more viscous, which is coherent. So we were doing exactly the same test as Julien, but at cell-cell interface, and being sure that the signaling was not a problem. So to conclude on this part, we were trying to shape the cell, trying to test the mechanics, trying to standardize single object. And by standardizing, this should remind you what people is trying to do in Landspuric using microfluidics, microchips, microwells, microstems. All of this is for systematizing the approach and having reproducible experiment. It's not only for fun that we do these kind of complicated approaches. And the conclusion here is, in fact, that T cell adapts mechanically at cell scale before the communication can be done by biochemistry. And why is it efficient? You have to polarize the T cell to perform the action. And if I'm here, and if I want to talk to Tatiana, which is in front of the stuff, if I throw something, or if I ask you to say something to the guy on the back, and then it will take some time. If I want the message to be very fast there, if I have a rope, I just simply do this, and the stuff climbs up very easily. So mechanically tuning at cell scale is one way to give a direction and the polarity to the cell. So if I go one step further, and this is the work mainly of Laurent and Keya, that I'm using to bridge the different scales, what we've seen also is that T cell can adapt to substrate. Yesterday, we've seen that if you tune the indent rigidity by tuning the red blood cell rigidity, then the cell reacts differently. Here, you have cells sitting on PDMS of different rigidities on a very large scale from, let's say, 100 kilopascal to megapascal, roughly. What you have is cells recognizing a substrate that is both activating and adherent. You have a ligand for the integrins, and also the ligand, you have an anti-CD3 for activation. And what you measure is the area of contact using interference microscopy. All in all, what you see, the stiffer, the larger the adhesion. This is what has been reported for a lot of different cell types. So this was not surprising. But if you remove the adhesion and only say that the cell adheres on something that is intrinsically activating. From monotonic, you get something that is biphasic. So removing the adhesion changed largely, very, very brutally, the behavior of the cell. And here the maximum occurs in a range that is typically the one of antigen-presenting cells, kilopascal, 10 kilopascal. This is the good range of DC, BC, macrophages, and so when they are activated. So why? The question is, in that case, that if you want to try to model it, you have, this is the work of Laurent and Keia for this modeling, you have to introduce effective bonds. Let's say one that is recognition plus adhesion and one that is recognition alone. And if you want to find a model that fits well the data, you have to introduce something that was non-trivial. That it's not only the outside part that may be different, but the coupling to the cytoskeleton to exert the forces that has to be different. So you need to introduce this link that is kind of a molecular clutch, something that, like what you have for changing wheels, changing speeds in the car, and is molecule dependent. It has to. That means that we need to know what's going inside the cell, what is the link between the recognition molecules and the cytoskeleton. But how do you look at this? So what we knew is that it's clear for integrin. Laurent presented that yesterday. Integrin have different shapes, more or less elongated. The more elongated they are, the more coupled to the cytoskeleton. In fact, there's an inside out and outside in signaling for integrin that is linked to this kind of tallinn-mediated link inside the cell. Potentially, some collaborators of us have seen interaction of the CD45. Again, this molecule that is in fact, so TCR is here, adhesion molecules are here, and CD45 is this one. So these are the large molecules. These ones, they have a lateral interaction with the cytoskeleton because they have a diffusion that is a bit modified compared to free ones. But it's not known for this. What is the interaction of these guys with actin that is just surrounding the immune synapse? The problem, if you want to study it, it's inside the cell. How do you probe the resistance of a bond that is behind the membrane? And in this game, again, a new tool. Laurent mentioned magnetic tweezer and acoustic tweezer yesterday, optical tweezers. Optical tweezers, they have a resolution of 1 piconewton when very easily like that. And what we did is we tried to use the bead with antibodies as a way to pull membrane tubes. Because if you are able to peel the membrane very delicately, that means that you are able to detach the membrane, or at least the bond with which you are pulling, from the cytoskeleton. And we observe, so here you have the bead, you have tethers here. So this is the idea. You touch, you pull, and eventually will rupture. You may have some tubes that look either like this. We've cooled it with rupture because it has a first peak, and then a quasi-horizontal plane. You pull at no force, just like flowing the membrane. This flow of membrane is reminiscent of the excess membrane that Julien presented yesterday when he was unfolding the microvilli and everything. Here you can have tubes without anything. You can have simply adhesion, or you can have no interaction at all. So our goal, it was to model this, and so we had a theoretician postdoc who worked on that. And we were to study these kind of things. So our model was a very simple maximum model, but we introduced the fact that the element values, the element resistance, could vary with time. For example, this one, K2, is the global mechanic. This one should be independent, whether you pull a membrane or not, because this is the cortex cytoskeleton. K1 will be the elasticity of the bond inside. If the bond breaks, K1 will drop. Here, the eta is in fact the equivalent viscosity of pulling the tube. But this will change because we will change curvature, and you will go from no tube to tube. So this will also drop when you have the stuff. And to take into account the fact that we have a spring outside, that is the one from the optical tweezer, we introduce the spring constant. And modeling this here, the one with the peak, we modeled it like this bond breaks, and then we pull the tube. Transition of the two parameters. And here, this one pulls the tube without breaking any bond, transition of only one parameter. And when you do this and fit all the values that you find, but in this part, in this very thin part, the values after the transition are the same for the same tube. So that means that this is coherent. So we have analytic and numerical solution for the model. I don't put the equations because they will be starting here and finishing somewhere there. So experimentally, we did something that we found clever. You can think what you found. So we tested different molecules at different positions of the immune synapse, using antibodies to have a strong bond. So this won't be the limiting factor. So we will be sure to test what's inside the cell. So we took anti-TCR, anti-CD45, and anti-integrin. This integrin is called LFA1, and anti-integrin with two different configurations. The one that can fish the molecules that are extended, that means bound to the cytoskeleton, or molecules that will fish the ones that are not extended, that means weakly bound to the cytoskeleton to get a control. And what we've seen is this. We measure, I just take here the K2 as the mechanic of the cell, and K1 as the mechanic of the bond inside the cell. What we've seen, you change the antibody, there is no much variation. This is good, that means that the cells here, they have the same mechanical properties. Here, that says that we start roughly from something where our resolution says it's similar. And then we put some drugs latrancolin A at very low dose to perturb a little bit the cytoskeleton. And by that, tuning down the bridging molecules, the bridging to the cytoskeleton. And what we observe, this is a good way of overcoming the limitation of p-values and everything. It's a way to look. In fact, this is the difference between this value, this one here, shows whether there's a significant difference between this and this. This one, the difference is here. If it's different from 0, that tells you it's a difference. What we've seen is that, first of all, when you add the drug, the cell is getting softer, and the same way for all of them. Perfect. The drug is there, and the drug is performing its action. And when we look at the bond, only the activated LFA1, that means the one strongly in interaction with the cytoskeleton, was perturbated, and also the TCR. So that means that we exactly saw what we wanted, is that there is some mechanical interaction of the TCR that is the same type of interaction that you may think for integrins. So it's the mechanism sensitive to drug at cell scale, but differentially at molecular scale, that is coherent with our understanding and our previous model of the interaction. Again, we play with the model. What is interesting, I just want to point out that this model can be adapted to different techniques because we have the spring of the technique into the model. So by tuning it, you can explore the same kind of interactions with different AFM, magnetic tweezer, or whatever. So all in all here, what do we have? We've shown that first, we have been able to probe an interaction inside of the cell. And second, that probing this interaction reveals that our previous modeling was right. There is differential interaction of the molecules at play during T-cell activation inside of the cell. So that's very coherent. But our methodology can be extended to other cellular systems. What is interesting is that having this kind of dual interaction, or let's say modulated interaction, is a way to probe the world for the cell to be more or less efficient, because you can pull. If the TCR is linked to the cytoskeleton, that means you have something that can help you pull on the bond and probe the bond, knowing whether the catch bond behavior at the very beginning could be relevant. If you don't pull, you don't test the catch bond. So all in all, it's coherent. The point now is, okay, we understand several steps. Single molecule, this mechanically interacting, cell mechanically understanding. How do you bridge the scales? And this is something that appears trivial, but for a physicist, where you step from one molecule to the first cell reaction is the most interesting, because you don't have all the non-linearities that signaling could introduce in the game. So we went for trying to bridge these scales. Let's say, study mechanically the early recognition of T cells in space and in time. How do you do that? Okay. Let's say in the lab, we have all of these tools, more or less. The only one we did not have was this one. This is a work that we made with Keia. And we set traction force microscopy that Gennaro is using. So here you already do some traction force microscopy, and this is simply something that you have around. But here we wanted to go at the very early moment of this cell recognition. Traditionally, people culture the cells and look at one time point. We wanted to see the cell performing the action. So we had to modify the existing protocols for two things. The first thing, make reproducible substrates. Super difficult when you've never been doing that. Always substrates and surface chemistry is a key point, and it's very difficult to maintain from lab to lab. People will give you a recipe and you are sure that you apply it strictly, it will fail. So, and second, we had to make it very, very soft, because you've seen, and I will explain again, but the cells, the T cells, they are very soft, they are 100 Pascal. The presenting cells, they are 100 to 1,000 Pascal. So you have to be in this range to be physiologically relevant. And so again, because you can see my small stickers on my computer, we are a bit open source guys. We went for Python and ImageJ solution. So everything had to be open source, just for the sake of being able, for example, to share it with you. If you want, you just simply take the code and you can apply it. This is the way science should be. So we did this and polyacrylamide gels, because these were very easily amenable to very soft. But the first difficulty was to have them doped with the right quantity of bits and then analyze the motion. So the first thing that we did is a little bit what we've been doing long ago with Yassab, who was present at Lance Biodit until recently. We made those gels and qualified them using atomic force microscopy, pressing with bits of the cell size, just to check the homogeneity. It was not only an average value, but also a laterally resolved value of the gel that we have. And we were able to make some gels in the range, all continuously in the range where the APCs are sitting. Typically, it's an order of magnitude lower than what is typically used in traction force microscopy, so it's more difficult to achieve. We checked that the stuff was homogeneous in terms of molecular coating using force microscopy, just to standardize again the experiment, because you will see one cell at a time, so you have to be sure that your conditions are at that scale reproducible. Then we studied, as we already did, is there any variation of the area as a function of the molecule, or let's say the gel elasticity, or the subtype of subcell? In fact, we worked for this stuff for two things. We use Jurkat T cells that are this model cell line, just to do the physics of it, because it's easier. And for the biology, we went for primary T cells that we were sorting with kits from human blood. So if you use the primary cell, it's more demanding, but so you won't do all the physics, but you can tune what subtype of cells you will use, whether they are memory, naive, activated, you can reactivate them and so on. So you have a way to play on the biology. On the contrary, with Jurkat, you can transfect them, you can label them, you can see many things with them. You can play with the object cell. So with the Jurkat, we've seen that if you tune the elastic modulus of the gel, you recover roughly the same behavior as on the previous study that we had with the PDMS gels, that was coherent. But most interestingly, if you change the subtype of the cell, they won't spread the same. And they will spread as a function of what they are supposed to do. Memory cells are the ones that you have after an infection. They should react rapidly and super efficiently. They were spreading more. That's fine. If you go for the elastic modulation of the cell as a response to the substrate, here you want to see they get stiffer as the software goes, the substrate goes. That's fine. But if you change the subtype on a given elasticity, also you will have differences. Why? Because they spread differently. So everything, the spreading and mechanics is really intrinsically coupled. And then, obviously, from that, we had the gel, we had the cells. Let's do traction force. Traction force, our way, you have a gel with bits. You let the cell sediment, and then the cell will push-pull on the substrate. You will move the bit, and if you're able to track the bit position, You can use the softness of the substrate as a spring to measure the lateral forces. What you have to see here, the main limitation is that you cannot see the vertical forces. You record the motion of the beads, you transform this motion of the bead into a map of forces or stresses, in fact, that you can recalculate from that the forces, actual forces exerted on each of the pixels of the map. And from that, you can do it on a movie, so you get time traces of the forces and the energy stored in the gel. If you tune the substrate from a non-specific, from a non-active antibody to an active antibody, the force increases when you have the positive one. So you have specific interaction from the start of this interaction. Most interestingly, time traces were not, when you were looking at them in details, we are not seeing something like always the same. Cells were having different patterns, mainly three families of interactions. One, trivial one, you encounter a surface and then you start to exert forces. The stronger one that we call sigmoid. Another one where the cell were trying to say, I touch, I pull, and then I release. I'm tired. Intermittent signal. And the last one was looking only like noise, but in fact it was not noise. If you were looking to the gel itself, the gel was having a level of fluctuation lower than this one. In fact, the cell was tiptoeing on it. And this is a behavior that have been seen long ago on solid substrate by our ex-boss, Pierre Bongrand, who described that as the first steps, the very early steps of the cell. First tiptoeing, then hesitating, and then engaging. And what you see is that if you put a non-specific antibody, you have no interaction, no active fluctuation. It's purely passive. Then you have a distribution of behaviors, and each of these conditions exert, as it's trivial when you see the curve, different forces, different energies. This one being the stronger, intermediate, and lower. What is fun is that these different patterns, distribution, change when you change the physics of the experiment. And the first stuff that we did is to change the elasticity of the substrate. We went for higher elasticity, just as what is commonly used in traction force microscopy. What appears here, look, the distribution change. You don't have this very strong forces system that is the sigmoid one. In fact, if you were to use the conditions that are non-physiologic, you lose the signal. So this is fun. This is why we had to go for the very low, very softest one. This is just a quantification, again. In fact, the force levels are similar, but the morphology, which contains the time behavior, are different, the morphology of the time traces. So using stiffer gels, impair initial forces. But more funnily, again, you want to label something. I see Gennaro likes to do any kind of labeling or whatever. If you label the stuff, you label the membrane to see where the cell is. You label the cytoskeleton to see the structure that they're pulling. And usually people in biology will say, that's fine. That's fine. The cell is the same. We can do mechanobiology with that. No worry. Look, Y-type jerkat with membrane labeled, jerkat with cytoskeleton labeled. Different distribution of time traces. Forces, they are similar. So if you were to look only statistically at the stuff, you would say, there is no effect. No, the effect is here. So force patterns are sensitive, while global readouts are not sensitive. And what is funny, now we go back, this is jerkat. Now we go to subtypes of cells, memory, naive, physiological cells. This is what we have. Forces are increasing the more you go from memory cells. This is normal. They have to react fast. They have to react strong to defend you. These are forces, these are energies. But what is really interesting is that it's a modulation. It's not a global force difference. It's a modulation of the time traces patterns. They react here, the memory T cell. This is sigmoid, almost entirely sigmoid. Sigmoid is, I respond. They have to respond fast. This is what you see. So these primary T cell forces are sensitive to T cell subtype, linked with their physiology and what they have to do, and linked with the biological function of the subtype. Conclusion of this, you can do early T cell recognition. We have different patterns. Some others, let's say Morgan Hughes group, where the student who did this work is now doing a postdoc. they've seen a lateral distribution of the forces that is different depending on the subtype. We have the time equivalent of their spatial distribution. Cells respond to the presentation, mechanics of the substrate, molecules that are presented, but and all this action is different for primary cells when you have different patterns like magnitudes. Relation to the role of the substrate, relation to calcium fluxes, we are trying to do these things at the moment. This will be the next story. And the take-home message is this one. T cells, immune cells, you can extend it to almost the cells you want, basically, but these are my favorite ones. They live a very physical, intense life. They are using forces from single molecule to single scale, cell scale. They can apply forces, they can feel forces, they can use forces for their action to defend us, and we can harness this kind of behavior. This is what Laurent tried to show you yesterday by saying that, for example, antibodies targeting cancer cells to be targeted by NK cells have to be characterized under forces, because this is a key point. These are the persons who did the work, the collaborators that we thank, especially Laurent and Kaya. We've been working together for at least the last decade. This is the lab and the funding situation. And I will just quickly point you to two things. We were at the conference in Cancun, International Conference for Cell Biology, and we represented the French Cell Biology Society. This society is open to everyone. That means you, as Mexican, can register and get the information and even get the help from the society for traveling, for example. This is very good, and also they have their own paper, Biology of the Cell, so where you can submit journals, let's say articles, reviews, and everything. And it has a good public, this stuff. Julien, for example, Julien Husson yesterday is publishing regularly in it because he says, okay, the community is responding well to these papers now, and they are well seen. The second point, so you can go on their site as BCF, you can enroll, there is no problem. The second stuff is, as Julien pointed out, we started a couple of years to try to federate, at least at French scale, and now at international scale, this immunobiophysics field. Over the last 20 years, more and more results have pointed in the direction that this is crucial for the research. So we have an online free seminar series. Every month, there is someone who talks. The talks are recorded for when the person allows us to record. And so you can see them. And so you can go on this site and register. And in 2021, the conference was fully virtual. Mexican people were there. 2023, the conference was in person, and Mexican people were there via our virtual hub. Please be there in 2025. And you can, if you can organize the hub at Landsby Audit, we could connect directly to one room like that. And so you could attend directly or see the talks. That would be a very good stuff. And also the persons, when they do this free seminar series, they're really open. They do very pedagogical teaching also at the same time. And to finish, we are here, as you mentioned, in the frame of this French to Mexican collaboration, where you can see UNAM is well represented, LAI and CINAM is the lab where Kaya is working, are represented, and Sorbonne, it's Mathieu. So basically, you have all the guys around in this small consortium. This consortium lasts until 2026, and we will try to push it further, because we think that it's a very interesting way for us to have an interaction, and so we can exchange our knowledge. And to finish, I will let you read this, for the one who recognize this small painting, except that these are T-cell labeled, and these are beads. Thank you for your attention. If you have questions, I will be happy to answer.
Well, thanks for sharing. Okay, good.
I'm sorry, just want to ask about this. The T cells which you're taking is T helper cells, right? Which you have taken.
Mainly these ones, the ones I've been using, Jarkat, they are helpers.
Jarkat, yes.
And the other one that we are using were helper ones also.
Okay.
Because they are more frequent. The CD8 also, it's a bit tricky to handle for us for the moment.
Okay. And Professor, the thing is, first of all, thanks for the great lecture. Thanks a lot for this because you have given a complete overview of and complete functionality of the T cells, how they are getting activated. That is most important. But my question here is, if we go to understand the mechanism of the whole process, like for example, the cytokines, I mean to say a B cell or any antigen presenting cells, they are approaching a T cell, for example, and the pre-activation is also there, right? I mean to say, of course, it's not. I'm going out of the scope of the study of yours, but then I think there is a kind of a cytokine and chemokine secretion, and then the T cells will be, they will be motivated to interact with antigen presenting cells, and then after that, once they interact, then after that, I think there is a kind of when you said activation, exactly. So what do you exactly mean by the word activation in this aspect?
So for me, this stuff is the activation when the cell will recognize the signal and start to, for example, go for calcium.
Yeah.
That's one step. In this frame, we totally forget about all the paracrine stuff.
Okay.
We just look at the simple contact-based model. And also, we very often look at only one of the three signals that are needed.
Yes.
Because we use very strong activation. So all of this, what I've said, mainly there's a bias potentially due to the NTCD3, because you shortcut one part of the mechanobiology of the stuff. Also, for the T and B cell, this is an ongoing project with Julien, I mentioned yesterday.
Yeah.
And we try to use B cell as presenting cells, which is not very often the way they are looked at. Very often people will look at the B cells the way I'm looking at T cells in their phagocytosis and then processing of the antigen.
Yes.
And so we are trying to reconstitute as much as possible of this chain of one activating, then going for presenting, then everything. But we will forget about all the guidance from chemistry because we are at single cell level. And don't forget that you should have the follicular dendritic cells, you should have all the epithelium and everything for guiding the stuff. And this is migration before. If you are a physicist, you try to simplify.
Yes, absolutely true. That's what, Thank you.
And also the problem is the more you go into this, the more you go into non-linearities of the system.
Yeah.
So it's more difficult then to model for us.
Exactly. Because that's what my idea was, that when you talk about the activation using the whole process, and one last question, what is the exact difference between when you say molecular level and the cellular level?
Molecular scale and cellular scales?
Yeah.
Molecular scales will be the kind of duration and scales that will be millisecond to second and a couple of nanometers. This is what Laurent showed yesterday with this small sketch.
Yeah.
This is the molecular scale. This is where you can do even artificially, you extract the molecules and you can make them together, pull at them and so on.
Okay.
Cellular scale, micrometer to 10 micrometer. Couple of seconds to minutes.
Okay.
In fact, I will stop after a couple of minutes.
Okay.
Because after that, the immune synapse comes in. And when the immune synapse comes in, as Jay said, its role is not yet clear.
Okay.
Is it something that ends up the activation? Or is it something where the activation is maintained? Is it something where the TCR is reused or whatever? This is not yet very, very clear.
Okay.
So cellular scale, and so basically if you say second, 100 nanometer to micrometer, this is typically where we are standing.
Okay. Yeah. Perfect. Thank you. Thanks a lot for this great lecture.
Thank you, Pierre. Yeah. And the signaling seems very complex, so very good. Good luck with that. Maybe you should look for some proteomic or transcriptome databases, and maybe Daniel can help you with the analysis to explore the signaling, because this is very complex. I think so. But maybe, or my question is, have you tried other inhibitors? I mean, to look for stiffness on the membrane, maybe cyclodextrins or nocodazole, or maybe look for the intermediate filaments, I don't know, in order to look for a cell component that is involved in this complex mechanics?
Thank you again, Aro, for this question, because the stuff we are starting to look at the membrane structure, but in the next project. In fact, one of our PhD students is working on the role of cholesterol and cholesterol export in this cell activation. So this would maybe help us to understand the role of the membrane, diffusivity, and so on. Cyclodextrin is dangerous because you don't only remove some of the lipids, you peel off everything, and it could be tricky. The other one, Cyto D, Bleb, and all these kind of drugs, we did not have time. This is something that we do gradually, but obviously, looking at the different elements of the cytoskeleton, it's also the point that I see. I totally understand. I totally agree, but their role could be at different time points. So the time dimension, that is usually the one that is forgotten when you do labelings and so on, is the one that will be the key for me, because you could have at one point the actin taking over for the mechanics, and then at one other time point you will need the microtubules. If you need, for example, to bring on the granzyme vesicles, microtubules, but at the same time you have to coordinate the actin to open and protect. So all of this, there's plenty of data in literature, but it's on different cell types, different systems, different experiments. And so it's very difficult to make a proper view of what could be the most probable. I don't say what could be the true, what could be the most probable picture. So that's something. For example, you could have the pH, you could have the Julien shown yesterday that host molecule that you generate by any kind of labeling could impair some mechanics. So it's complicated, as we say.
Another question? Well, let me talk that the Dr. Pierre is the artist of image that we represent, no, that represents our Congress. Pequeño.
I just cut the presentation.
El image, el logo.
Up. This one, which one? The last one?
No.
The conference logo.
Oh.
Wait. I'm just speeding. There's even slides that you did not see. This one?
No.
This conference logo. I designed it, yeah, sorry. I'm a bit tired, I would say. And I'm changing my languages 5 times a day. Yeah, just pick up this. It goes on the screen, I think. Let's say technology is supposed to make your life easier. Just a comment. No. So basically, if you go and see the logo, there's one very important stuff to look at. you have one red cell that is in fact endangered, so it's fact that is red. There's a green cell, the red one, the round one, that is in fact the immune cell, scouting for the danger. And the important stuff is that you have two small things that are looking like this. And this, if you go to the Museum of Archaeology, and if you look back at the entrance, you turn yourself like this, and when you look on the front wall, you will see that this is the symbol for discussion, for the voice that the Aztecas and Mexicas were using back on the time on the codices. This is very funny to mix up this kind of skills and this kind of stuff. So the techniques is always espionage. The computer is dead. ¿Qué quieres hacer? Necesito bajar la resolución. Ah, por eso. No sé si va a funcionar, ¿no? No sé si se ve algo. Changing computers, changing brands, 3 different brands of computer. I have to say the one that it was the best working was under Linux. This was mine, again. I'm just advertising briefly. Use Linux, no problems. Even macOS is not surviving. Look at it. Anti-GAFAM propaganda.
Stop.
And now we will try with a Google computer again. I'm sorry, I could not resist this kind of bad jokes. Here, look on the screen here, the small volutes of the discussion. So basically the design is very simple. I took back some colors that were from the LibreOffice conference last year in UNAM. And these colors are the ones that you will find, for example, on the paintings of Teotihuacan, or on any kind of painting that are coming from the remains of the Mexicas. And then this circular structure, something that looks a bit like the Piedra del Sol, with this periodicity of the motion. And in the middle, you have what we are looking at. It's there. This small interaction between immune cells and tissue cells. This is the theme of our Franco-Mexican interaction. And obviously, this has been stolen using open source softwares from the Museum of Anthropology, directly from the codices with a picture. So that's fine.
Okay, thanks.
It's normally stopped.
Bueno, ¿qué onda, Matt? ¿Nos escuchas?
Sí, los escucho. ¿Ustedes?
Ah, muy bien. Ya, están ajustando algunos detalles.
Ok. ¿Comparto la pantalla ya o no?
No, es que tenemos problemas de conexión ahorita con el proyector, pero ya casi.
Ok.
Bien, bien. Hola, ¿ya?
Hola.
Ok, bueno,
Sí.
hoy vamos a presentar ahora la presentación de Matthew Oudfoy. Él es un gran amigo y fundador también del Laboratorio Nacional. Pues él llegó a México para haciendo una estancia, haciendo su postdoc. Él estudió su licenciatura y maestría en Ingeniería Electrónica en Grenoble y después hizo su doctorado en Irlanda. Estuvo aquí en la universidad formando a muchos estudiantes, dando muchos cursos y muchos de nosotros fuimos parte de su equipo. Formó el Laboratorio Nacional y ahora se encuentra en la Universidad de Sorbonne y en el Laboratorio de Biología del Desarrollo. Su trabajo se centra en el desarrollo de modelos biomiméticos para cultivo celular y especialmente en plataformas microfluídicas de tipo órgano en chip, donde pues le interesa mucho la mecanobiología en microentornos fisiológicos, los modelos de enfermedades hepáticas y modelos físicos de morfogénesis celular. Entonces, él nos acompaña ahora con su plática titulada Strategies of Microvessels Engineering for Vascularized 3D Culture Platforms. Bueno, él lo dirá mejor. Y pues, sin más, ahí está una breve introducción, Matt.
Gracias, Aaron.
Y pues bueno, vas.
¿Ya me escuchan?
Sí, súper bien.
¿Y ven mi pantalla? ¿Sí?
Sí, es la presentación, ¿no?
Perfecto. Sí.
Muy bien.
Pues muchas gracias por haberme invitado. I will switch to English now because I would like to do it in Spanish, but I think all the talks are in English. So the outline of my presentation is basically divided in 4 parts. The first one, I'll give you a few generalities on integration of why we need many signals for cells to feel at home, if you want, for new in vitro models. And I will present 2 types of in vitro models, which are basically lookalikes. One of them are the organ on chips. I'll give a brief introduction of their history and why we use them. And I'll switch to what we now call MPS because, to be honest, organ on chip is a term that scares a little bit people in the society. So it's better to say microphysiological systems, which actually also means a bit more about what we want to do, which is mimic what you find in vivo to have better in vitro models. Then I'll tell you more about why we want to insist on vessels in those organ on chips or MPS, and how we can construct those vessels and why physics is also everywhere in this construction of vessels. I'd like to finish my talk with presenting the last results of the team that I started 3 years ago here at Sorbonne in Paris. And if I have time, I can detail the paper that we've published, but the paper is published, so it's easier for you to check it online also. And you can ask me always if you have questions in the future, because as usual, I will probably run out of time. So please, Aaron, stop me if I extend too much. So the general aim of this organ on chip and MPS philosophy, if you want, is that you want to integrate in vitro all the known in situ signals that are impacting the cells, that are defining what a cell is, all the Q markers, the phenotypic markers that define its function, its shape also. And of course, if it's part of an epithelial, for instance, layer, the shape of the whole tissue. So usually in the past centuries, I'd say, since we know how to culture cells, we were focusing a lot on what's the good medium, what are the good nutrients, how do we need to give the right chemical signals to the cells so that they receive what they need to be what they should be. But now we know that geometry, architecture that surrounds the cells, the composition of the proteins that are surrounding them also, and the mechanics, including the dynamic cells that are dividing around them, are very important cues for the cells to be what they are. They have a lot of receptors that are able to receive those signals and to tell the cell what it should be. So tissue shape and functions of cells and tissues are emerging from this multiplicity, if you want, of all the environmental stimuli that are important to mimic. The inside of the cell now we know is kind of a tensegrity structure where you have tensions and compression, which I think you've heard a lot about in the past days and probably again tomorrow. But I want to emphasize on the fact that they also are shaped by what's around them, and that's what we need to control if we want to engineer the right environment. Unfortunately, in vitro, the culture systems, the traditional culture platforms are infinite if you compare to the scale of a cell. They're very uniform, very flat, also very stiff. If you know a little bit about the elasticity or Young's modulus that Pierre-Henri was talking about earlier, of a tissue, it's in the kilopascal range, but when you use a plastic or glass, it's in a gigapascal range, which is way beyond what you would expect for a cell. And also it's very static, usually. You know, you change the medium every now and then, but it's very boring for the cell. So you usually have a 2D culture system that's boring, and the cell doesn't look like it should be. And you lack a lot of markers, which I painted here in colors, and they're not there. So the cell is not representing what you want to study. So a lot has been done in the past to improve those platforms. Mainly, you will hear a lot about 3D culture systems, where cells are placed in soft hydrogels. They're organizing themselves in spheroids. If you use tumor cells, in tumoroids. If you use pluripotent stem cells, in organoids. But they're basically trying to organize the way they can using the physics that they have around them and the signals that they receive, and most of them are lacking still. But it's improving a lot, the platforms that we are using for in vitro culture. It's improving the identity of the cells and the physiology, and it's also improving the models that we have. But organ on chips have made another breakthrough, because they are combining many techniques that I will not detail today. One of them being microfluidic technology. So that's where I come from, the microfabrication systems, where you can use transparent plastics, glasses, or whatever substrate is useful for you. So you can still image the same way you would in traditional culture platforms like those that were developed for that. You can interrogate your cells, but the better tissue model that you have in your hand increases the fidelity, so the reliability of the model. It's closer to the in vivo, but also the long term, because the cells are behaving the way you want for up to weeks and even months in some examples. So that's very good for drug metabolism testing, let's say drug testing in general, even toxicological evaluations. So the models are very useful now in pharma, and you will see in one or two slides that the regulations are changing in some countries. In Europe, it's discussed. I don't know in Mexico, to be honest, at the moment. But also, and what we are interested in our team is that if there are better models, then we can also make more fundamental research, look at mechanisms, what's happening really in vivo, how a drug can affect the tissue, how a lot of things that are happening in physiology or pathology are happening, because we can have more reliable models. So from 2005, in the US, the FDA has funding a lot of organ-on-chip research in many different universities and public and private labs. It became a disruptive technology. I was happy to say, since in 2019, National Geographic helped us to tell our families what we were working on, because it was part of a very nice article where they said it's possibly a revolution in medicine. So you know about revolutions, they take time to actually settle, they take time to be a reality in our daily lives. But now we have technological companies in the US and Asia and Europe, probably now in Mexico too. The Lance Biolite is pushing for that. But really, they rely on a very important part that I will insist upon now, which is co-culturing of different types of cells, where you usually have an epithelial layer and an endothelial layer. The endothelial cells are the ones that are lining the vessels. They are the ones responsible for the barrier functions of those vessels that you have here, allowing some things to pass and some not. Like neutrophils, for instance. There is a very nice video that you can find online where you have the infection of a lung organ-on-chip system that's recorded on camera, that's live, and you can see, I don't think there were neutrophils on this video, but there were immune cells phagocyting bacteria that were part of the infection that they could simulate on an organ-on-chip system. But you can also flush, if you want, in this blood-like channel that's here below, a lot of chemicals, cytokines, whatever you want, and look at the response of the cells that are, I insist, looking like what you would find in vivo. So in the lung on chip, again, they were able to mimic an edema on the chip to make those cells produce some fluid and then test drugs, anti-edema, if you want. So in this type of engineering process here, where you have multiple layers, and I'm sure you can visit the Landsbiodit facilities and have a look at how they fabricate this, which takes time to, like in material science and engineering, in biology, in chemistry, in physics, in mathematics, a lot of fields have to be done, but basically what you want to mimic is this. So why are endothelial cells and vessels important? What do they do in those models? So for that, I need to go back a little bit and talk about blood vessels. You know about their transport functions. I told you already about that also. It's bidirectional, O2 and nutrients in the artery system. And then this system connects to the vein system through capillaries. And as you go in this connection here, that's the part we're interested in. I hope you see my pointer around the capillaries, where you have thin layers here, only one cell thin blood vessels, compared to the thicker layers that are just made for transport, not for diffusion. So the capillary layer, that's where everything passes. The transport happens here, and then you have the interaction with the tissue. So all these small, tiny vessels, which you cannot see here in this image, are the ones responsible for connection. So you have a highly hierarchical organization, few large and stiff vessels, branching down to many small and soft, tiny vessels, which are called capillary. And they're very tiny. Look at the scale here, like 10 micron, 9 microns here. And they're almost not protected by anything. Sometimes you find pericytes, not everywhere. You have a basement membrane that's very thin, but they are made to diffuse what's going through them, and from the outside in and from the inside out. And they're very organized and they're very dynamic, and they can remodel also, which is very important for what we want to do. So those capillaries are small. Their thickness is very thin, as I said, but they handle quite large pressures if you consider the size of them, just for flow to happen, just for exchange to happen. And what's very interesting also is the way they form. Not a lot is known about them. This is an example from 2007, and we still don't really know exactly how those single cells here can really organize as a vessel, then capillary plexus that will then connect to the heart and make this fully recirculation system with new vessels here. And that's what we were interested in our team. That's what we want to build, to try to instruct cells to actually form new vessels. So you have two ways of forming new vessels. The first one is called vasculogenesis. It's also called de novo, new vessel formation. So basically you had no existing vessel, no pre-existing vessel, and some cells are organizing themselves, self-organizing, we say sometimes, to form these nice cords here, which still have no lumen, if you look at them here. And then eventually they will open a lumen here, okay? And then that's where the blood will flow. But then another process also exists, which is it's studied a lot in vitro. It's called angiogenesis. And this one comes from an existing blood vessel, which is represented here. Blood is still flowing inside. And for some reason, many reasons, one of them being a tissue needs more access to oxygen or more nutrients, but also regeneration, repairing of the tissues. You will have a protuberance here growing and making a new vessel that needs to avoid leakage, if you want. So you need to create a new tubing system. And think about plumbery. If you want to connect tubes together, it's very difficult to avoid leakage. And that's what you need to do. So angiogenesis is also a very complex, not completely understood process that needs in vitro models to be better understood. And I will finish this very long introduction by saying that the life cycle of the vasculature, which is what I've summarized very briefly for you, is something that's not really well known. But what also needs to be studied is the specificity of the endothelium, if you want, in terms of functions, but in terms also of tissue specificity. Like each tissue will have its own capillaries, if you want, and even in multicellularity, the pericytes that go around, the fibroblasts that communicate with the vessels, all the epithelial cells, of course, that communicate with those vessels. They have their own specificity, and you cannot take a vessel from a tissue and take it from in another tissue and find the same barrier function, cell trafficking. Everything is very well controlled at the tissue level. So that's why we don't really understand how it works, because each tissue will be different. So it's a very long process, but still we made progress as a community, as a scientific community, in building vessels on chip. So there are multiple ways to build a better endothelium, a better way of fabricating vessels for organ on chip and MPS. The first one I showed you is a flat monolayer, as you can see here. We use microfluidics, and due to the, let's say, the microfabrication processes that we use that come from engineering of silicon chips, which is almost layer by layer, so not really 3D. That's why you have flat channels, flat membranes. It's always flat if you want. So the cells, when you seed them, they will fall by gravity, even if you flow them, if you flush them, and they will end up adhering to a flat substrate. We call them monolayers, those architectures that they form, like a carpet of cells, if you want. But the fact that we use those monolayers, like they look like 2D, under a flow, improved a lot the phenotype of those cells, because they need the flow to be endothelial cells. That's something we understood 20 to 30 years ago. They need to sense this flow rate. You know, if you go a little bit about the physics of those flow rates, they can mechanically sense that there is a stress, and we call it in English the shear stress. Okay, so that's the shear that makes them be endothelial cells, if you want. You need the right amount of stress. If you put too much, you will stress them. If you don't put at all, you will lose the phenotype. So you can control the geometry of the chip that you use, the thickness or the depth, if you want, the width and the length. And the shear stress depends on the geometry, which is very important. So you can even control the geometries that you want and increase or decrease the shear stress. Here you have a larger, wider channel, so you have a lower shear stress. And here it's narrow, so you have a higher shear stress. And the cells you will see in this type of experiments, this was published 10 years ago, cells do not behave the same way, depending on the amount of shear stress that you apply on them. You can also add up a new way of culturing cells inside microfluidic channels by adding another type of flow upon them. That's what we're doing in the lab at the moment. You embed those cells, endothelial cells, inside a gel, and you apply a flow inside this gel like you would apply it through a sponge. It's a porous gel, so the flow will not be like this one, like a fluid flow, like a constant fluid flow inside a tube, which you have seen probably through a tube. This one is through a sponge, through the pores, so the flow is much lower. It's called interstitial flow for a reason. In the interstices, it can go. But still, we know now that those cells can feel this flow. And that's what happens in a tissue. You don't have a hollow channel except in the blood vessels. Otherwise, you always have extracellular matrix. So you can even replace this gel by extracellular matrix, which looks even closer to what you have in vivo. And you can try to study how those cells react to this type of flow. And that's what we are doing at the moment, because it seems to be very important in the way they behave and in their phenotype. You can also add more to that. You can, if you have the chip, apply hydrostatic pressure many ways, like you put a weight that you can control and you know exactly how much pressure you apply. Or you can use the neighboring channels nearby to put some more pressure to the sides, and you can squeeze the cells inside the gel. So flow plus pressure inside. And of course, as you can see in the lung-on-a-chip system, you can use your side channels to your benefit to actually create a vacuum inside the channels, like you would pull when you aspirate on a straw and you put your finger on the other side of the straw and you see the straw collapsing as a vacuum. So they do exactly that to the side channels. And because all the rest is a flexible silicon material, which is PDMS in this case, it extends, and you can create some tensile stretch, which is also something that cells, some kind of stress that cells will suffer in vivo. So you can do a lot of different aggression, if you want to look at them that way, or stimuli that are important for the cells, and study how cells are responding to it. But if I go back to this one, we still have monolayers, and the vessels are tubes. I showed you they are tubes, even the capillaries. So we're using monolayers, and of course I could cut this tube and flatten it. It's known now that the curvature is very important. It's a signal also for cells. So it's a signal mechanically because of whatever happens inside and outside of those tubes. And I invite you to check this review that was published by Abdul Barakat's team in the same lab as Julien Husson that you saw yesterday. A lot of those signals, topography, shear stress, pressure, tensile strain, and curvature are very important for cells to be who they need to be. For instance, just the curvature. If you look at those two, this would be more an artery-like vessel, and this would be more like almost capillary-like vessel. And it's known that it's important, this curvature is important for the cells to behave, to acquire the phenotype that you expect. You can't have a capillary cell be in a large vessel. It will switch to a larger vessel type. The specificity will change. But also the stiffness and the strain are important. So I need a tube. And that's why another technique has been developed where you use a gel in this case. So it's not a chip like this with channels. It's a channel that you create inside the gel. And I help you with this image here of a jello. So because you do an ECM hydrogel or any type of hydrogel you want, imagine when you prepare this jello inside your fridge, you first have it as a liquid and you mold it inside the liquid, inside the mold, so you have the liquid. But imagine now that before you prepare that and you put it in the fridge, you place a spaghetti inside, not a cooked spaghetti, a stiff spaghetti. So you will go inside and the liquid will go around, it will conform around the spaghetti, and then you put it in the fridge. When you take it out, it cross-linked, it became a gel. You can pull the spaghetti out, and what you're left with is a hollow channel like this one. And this hollow channel, you can then populate it with the cells the same way you would populate a flat channel here. The cells will go around wherever they can adhere. Of course, they will flow through. Some of them will, but most of them will adhere to the side because it's ECM. They have the ligands to recognize those, they have the receptors, sorry, to recognize those ligands, and they adhere, and then you have a tube. So I have a tubular monolayer in a pre-existing tube. And then I can have a luminal flow inside. I can control the luminal flow with the pumps or whatever I use to control the flow. And I have a more high fidelity model. It's a tube now. Of course, we don't use spaghetti. Spaghetti is too big. We use acupuncture needles. You can find the right caliber of those acupuncture needles. They go down to 150 microns. Apparently, we didn't find anyone that was smaller than that. And again, in Lance Biodit, they know how to do that. You can approach them and ask them how to do it. And the cells are seated perfectly inside. But still, as I wanted to show with this meme, it's still very big for capillaries. And you will see again for the applications we have in mind, they're embedded in a gel. And sometimes it's not the best. So everything we've done now until now, we thought that form dictates functions, as I said, as much as the other stimuli that we keep, of course, in organ on chip systems. But shapes are not fixed in vivo. And when we have the natural shape that we want to mimic in vitro, and we make a mold out of it, we want to imitate it. We know the shape, we know the cells. We place the different cell types inside this mold. We realize after a while that the cells are changing their phenotype, they are transdifferentiating, they are not adapted for a long time inside their molds. And that's because cells are not like Play-Doh. We need to allow them to construct their own structures. Yes, form can dictate functions, but you need the form to be shaped by the cells themselves. They're active, they know how to make those, and the vessels are a good examples for that. So that's the third strategy that you can find in the literature, where you allow them to self-assemble, to self-structure, to self-organize. So you take the same gel that you have here, but you place the cells individually at the right density, meaning at the right distance. And because they're adhering, they have ligands, again, to which they can connect through their receptors. They will pull on that gel, and provided the gel is made of the right ECM, the right extracellular matrix, and at the right mechanical properties, they will start to attract themselves, connect with each other, polarize, migrate, and form spontaneously what we call self-organized vessels. They emerge spontaneously in 5 to 7 days. And what's really amazing, it's because it's a gel, you can create this interstitial flow I showed you before, thanks to a well-designed chip around that channel where you have the gel. And you will see that they luminize, meaning they open the lumen, exactly like they would do. Well, exactly, we don't know. Actually, we haven't proven that's what happens in vivo, but it looks exactly like we would find in vivo. They open the lumen and they become perfusable, and they actually become perfused. And I don't know exactly because I couldn't see the talk of Milica Radicic the other day. I will definitely check it online. But I think she showed you that when you mix them with macrophages and fibroblasts, those vessels become more tissue specific. You can get a more phenotypical cue that looks like the tissue you want to mimic in vitro, which is very important for you to remember. And now where it's going is that you can interface those vessels with other tissues, like, for instance, tumoroids, 3D tissues that you place inside the gel or on top of the gel, and you will find that you can guide the cells to interconnect with those tissues and vascularize them. So you recreate what's happening in vivo in a way. Of course, a lot has to be studied to exactly find the mechanisms and to understand if it's closer to what happens in vivo. But people are doing models like that, like in our lab, Bruna Safi, who is a PhD student, who will be visiting Mexico next year, has designed this type of chip where she loads a gel with endothelial cells. She uses UVEX in this case. She applies the right pressure difference across the gel to form the luminized vessels. And our goal is to interface that with kidney tumors, because we know now that some tumors are instructing vessels to form very aberrant structures, which are making all the drugs we want to use inefficient. And we don't know the mechanism of this aberrant structure formation, and she wants to mimic that in vitro to then look at how the drugs are affecting or not affecting the structure of the vessels. We also use the same chip. Wenjin Xiao is a new researcher in my team, was just recruited in October. She uses the same chip with liver spheroids. So she obtains the liver spheroids from a collaborator at the hospital, directly from the patients. The spheroids are made out of parenchymal and non-parenchymal cells, and they make spheroids. They've been making spheroids for a long time for drug testings, and they actually sell that model to industry. But what Wenjin was saying is that we should apply a flow on those spheroids and see if they don't self-organize. You still have some endothelial cells inside those spheroids. Can we self-organize them by just applying a flow? And this chip was actually made thanks to a mold that the people at the Landsbiodit shipped to us. So inside this mold, you have basically tiny wells of 300 micron diameter, and that's precisely the size of the spheroids that we culture in those ultra-low attachment plates. And when you place them under flow for 3 days, you see a difference, which is very notable. This is one preliminary result that she found. No flow, static condition, versus flow condition. So these are two different spheroids from the same patient. After 5 days, you can see that the vessels are organizing, and she could even find in confocal microscopy some opening of lumen. So now she will place those spheroids on top of the structures that Bruna is fabricating. Is actually, inside the chip where the cells are guided to fabricate their own vessels, and see if we can anastomose, which is a medical term to say that we connect the two vessels, those that are inside the spheroid and those that are outside, and see if we can vascularize this liver spheroid. Of course, that's to mimic a liver drug testing capability. But now I want to step back a little and tell you another thing we wanted to do. I think I still have time. I have a few minutes left. The liver capillaries are called liver sinusoids, and they are very different to other capillaries in the sense that they have almost no basement membrane, almost no protection around, if you want, almost no capping around the sinusoids, because they have to be very porous. It's almost a porous system if you model the liver. We published something on that a few years ago. It's very important that you imagine it's not embedded in anything. Actually, if it's embedded, it's because your liver is sick. It's because these cells here in blue, which are almost like the pericytes, but they're also like fibroblasts, if you want. They are supposed to help repair and produce extracellular matrix. But when you have fibrosis, and I'm simplifying a lot, I'm oversimplifying, but they do produce some extracellular matrix around, and that stresses the endothelial cells, and you don't have healthy endothelial cells. So we don't want the embedded structure for liver sinusoids, because that would not mimic what you have in vivo. And we want to also mimic how the blood flow is impacting the phenotype of those cells, and how maybe it can trigger some pathologies, or it can trigger also liver regeneration. So we need a self-sustained, if you want, not embedded vessel to do that. And that's precisely what we published. So that's Jimena's paper, I would like to say, because that's her PhD work. She will defend very soon at UNAM, that it has been reviewed. It was just published last month. You can check the paper. I can send it to you if you if you don't have time to flash the QR code now. But we are very proud to show that she could guide endothelial cells to form a vessel. And how did she do that? She came back to developmental biology and the physics of forms and shape to try to understand how vasculogenesis can be controlled and engineered. There's a nice essay that's known for a long time now that if you place endothelial cells on Matrigel or Geltrex, which is another presentation, another brand of the same extracellular matrix, which is very soft and very rich in laminin, if you place those cells on top of Matrigel, they will form these tiny cores that some people call vessel. And it's been published in 2020 that it's just because cells are pulling on their substrate, thanks to their focal adhesions, thanks to their integrin to ligand connections. They're pulling on the substrate and they start polarizing, changing their shapes, migrating towards each other, and form networks that you can see here in this animation. So it's the cell tension in the ECM that drives the cord formation. And another paper was published at the same time in Cell Reports, where they showed that if you do that on collagen, the reason why it works on Matrigel or Geltrex and not on other ECM is because Matrigel and Geltrex have a lot of laminin, and laminin is more plastic than elastic. And here, just a little bit of physics. Elastic means that when you apply a deformation because you push on it or you pull on the material, when you remove the force, immediately it goes back to its original form, original shape, original structure. When it's plastic or when it's viscoelastic, it takes more time to go back. When it's plastic, it actually doesn't go back. So you deform the material, and after you finish pulling on it, you remove the force, and it stays exactly in the way you pull on it. So those, I don't know if you see them, gray or purple lines, are like chains of force that remain in the matrix, in the matrigel or the Geltrex. So it's like a shear stress that cells will feel underneath and to which they align. And you avoid a monolayer thanks to that, because the laminin plasticity guides the cells toward this network formation. So it's definitely a physical mechanism that happens. So you can basically do exactly that with the endothelial cells from the liver, which are called LSECs, liver sinusoidal endothelial cells. She tested murine cell line, human cell line, primary cells, thanks to Genaro and his team. And many different endothelial cells actually were tested, and they all work very well. But if you wait longer, if they form those cores, they actually never luminize. So we stopped calling them vessels because they have no lumen. And after a few days, they contract and end up being spheres. Like we even call them apoptotic bodies because we think they're dying. So the hypothesis we made at that moment is that there are no preferential orientation. So all those cores are pulling and pulling and pulling and pulling, and the cells end up, collapsing into spheres. So we tried to give them a preferential direction, anisotropic direction, if you want. And when you do that between two PDMS slabs, so you have the Geltrex in between, you can find longer sustained structures, like this one, above a certain critical size. So here, you will never actually reach the critical size, so neither of those lines is actually sustained for long. But when you do that, they're sustained for long, actually up to weeks, if you want. They're still living, they're very thick. But what Jimena found is that when they do that, they also pull on the ECM and they bend the ECM inside. You see here? They fold the gel inwards. So you can find in literature that you just have to place some matrigel on top and they will polarize and make a tube, which is great because then you have a tube with the lumen. But it's not great for us because it's embedded in the gel again. And that's exactly what we want to avoid. So how can we make these sustained lines actually fold, but without being embedded? We have a half tube here. Like if you want cells are pulling on the soft substrate here, that's what you would do on a soft fabric. I help you with this, this guide. And you fold the gel inside just by pulling, okay? That's what they do. So it's not locally that they fold, it's because on a large scale, they fold the material. So we were thinking, can we fold the material that's actually folding on top of itself and obtain a sustained, long-lasting tube like the one you see here? For that, Jimena used anisotropic structure around a dome of this laminin-rich, which is Geltrex in our case, droplet. We have a very long micro pattern, very thin but very long around the dome. And what she found is that the center of this dome, cells would form cords, which is exactly what happens on top of matrigel. On the outside of the dome, which is glass, a monolayer will form slowly but surely. And around it, she found that she has a very thin layer of laminin, which I will show you in the next slide, which is exactly like what happens with the coffee stain effect, which is called Marangoni effect, where because of the evaporation that's faster at the edge than here, where it's thicker, you have more of material that concentrates here, like the coffee particles at the edge of this stain. And laminin does exactly that in the Geltrex dome or matrigel dome. You have evaporation, you have a strain here, a movement here. And if you look at the middle near edge or edge density of the laminin, it's very, very dense, and collagen 4 also. They are almost completely recruited here just at the edge, like the coffee stain effect. So you don't have a stain, you don't have a dry droplet, it's still a droplet, but she managed to cross-link it at the right conditions to get a laminin pattern here. And cells love this pattern and will align along this pattern. And here around this, they will form a cord that will end up being a tube. And when you look more closely, they actually fold the gel at the edge of this droplet. Here they end up after day 4 as a luminized tube that sustains. Here you see a more roundish vessel. It's day 8, and we have found this lumen that is only 5 to 10 micron in size, which is basically the size of a liver sinusoid, just at the edge of the droplet, almost not embedded. And actually, we can detach it, so it's not embedded in the end. You will see pictures up to 15 days. So that's pretty cool. When we look at it under an electron microscope, I will go fast now. The monolayer doesn't look at all like, well, the face of the cells, they're telling us we're not endothelial cells, basically. They're more like an epithelial monolayer, if you want. The cords are pooled, and they're more cords than luminized cells. But the luminized vessel is basically like curved like a vessel. You can not see the scale. I'm sorry, I should... yeah, it's pretty bad. I didn't put the scale here, but this is 10 microns, okay? So you have the vessel here, it's rounded, and if you look inside, you have a lumen. And if you look more closely, this is actually after 10 days, but just a few days before they started secreting their own basement membrane that you see here, some fenestrations could be seen. And we have quantified their amount, and they're actually visible and present here for 5 to 7 days, which is a very important hallmark, telling us, I am a liver sinusoidal endothelial cell, which is very important. The way they do it, I have a short animation for you. They first have a migration stage towards the edge, and then they really like this edge, and they never move from this edge. They form a train 1 cell wide, 1 cell thin, if you want, of a structure. And after they completely go around the dome, they stop their polarization in this direction, and they start dividing vertically, like climbing the dome, if you want. And by doing that, they end up forming a thicker train, and they pull on the substrate, exactly like they were doing when I showed you they were folding the gel, but now the gel is vertical, and they displace this cord, and they completely end up blanketing their structure. If you allow, I will skip those slides where I give all the quantifications Jimena has done about the approach phase and the folding phase, all these angles that she's measured over a wide range of samples. You can have a look in the paper just for the sake of time. Just one very quick message here, that we found that exactly at the hinge cell where the folding will occur, we have a depletion of ECM that we have found very consistently. So ECM is disappearing from there, and that's where we found, they're pulling, so it's getting thinner and thinner. And here you see the intensity of the ECM just at the hinge cell. So the idea that Jimena had to prove that they were actually pulling, like when I showed you the image of the 2 hands pulling of a soft fabric, she used fluorescent beads inside the dome. These are the cells in green. And she took many, many videos to show that when you look at the displacement of the beads, I will skip this one and go directly here. Basically, they're doing exactly this. They're pulling along the gel. So we're seeing the dome from the top. And when they pull on that, they fold the gel like this, like a wave, if you want. They fold the gel upon each other, and they're pulling their blanket of ECM, a very thin blanket of ECM. And when they do that, in biology, you know that if you have laminin everywhere around you, which is visible in this image here. Very nice image. The cells are inside, and you have laminin and collagen 4 around here, mainly laminin. Okay, remember the pattern. You have laminin everywhere, so this is signal for cells of basal. So the podocalaxin signals goes from stage 3 to being basal here to becoming apical here. So it's switching its position, it's changing its location from being basal to being apical. And that's exactly the signal that cells were lacking to open a lumen here. And one day after this stage 4, one day after that group, the blanket that they were pulling on top of them, they opened the lumen, and we found the lumen consistent. And it also correlated with the maturation of the tube, where you see here she quantified the condensates of 0.1 per stage, per cell. Stage 3, when you already saw a tube-ish structure, 0.1 was not organized. And at stage 4, which is this stage, after podocalaxin went apical, you definitely have a more structured vessel. And after that, one day after, the vessel that was finally visible here in electron microscopy became more rough, if you want, in a sense. And you had the line bundles, wavy bundles of extracellular matrix, basically collagen 4 and laminin, which she quantified also. And you can see here, you can believe me, that this vessel may look like it's embedded, but it's a very thin vessel, and it's only its basement membrane that you have access to here, and you can detach it. And here's a nice picture of it also. It looks like this one, the experiment we made with the lycra, which I found very interesting to show. So here is the summary of the process. You can find a way to have a micro pattern of laminin at the edge of the dome that cells will find, if you want, if they're less than 50 micron away from that edge. The rest will form a monolayer. They will align along this edge, proliferate a lot, exactly like they would form a new vessel in vivo. And after they completely go around the vessel, the dome, they start proliferating away from this train of one cell and pull on the gel to finally form this nicely organized and polarized set of cells, maturing the tight junction and making your cell a tube. So I finish with this by acknowledging the whole team. We are a new team now, and Jimena is still part of it in a way, because she hasn't defended her PhD, but she's already working in a different team now with experts in immunity, so closer to you, Pierre-Henri. And of course, all the collaborators we are working with, because this vessel is not only for the liver anymore. Now we are building more vessels for different tissues, and we are going towards a multi-organ-on-chip system that we hope will be connected with this kind of tube. So with that, I thank you for your attention, and I'm ready for questions if you have any. Thank you. And of course, I insist that if you have more questions in the future, you can email me whenever you want.
Okay. Someone have any question? Okay. ¿Sí? A ver, ahí va.
Hello, Matthew. Thanks so much. Do you think that in the long period you can disattach the vessels that have been formed in the dome and then analyze them maybe in a singular way?
Yeah, it's a very good question. That's what we're doing. I don't know if you're seeing my slides still.
Gracias. Again, hello, Matthew. Thank you so much.
I heard you.
Do you hear me?
Yeah, I heard you.
Yeah, thank you.
Do you see my slides? I placed an extra slide here. This is a detached vessel that's 2 centimeters long.
Nice. So isolated from the dome.
Yeah, you can cut it very easily. It's actually amazing when Jimena was thinking that the monolayer next to it was annoying us, that the cells would be attached to it. Actually, when you prepare for immunostaining, for any confocal imaging or any any staining that you want, the monolayer detaches very easily, and you always find that the tube is very, very well structured and very stable. And we were able to detach it. And the only thing we haven't been able to do now is to perfuse it correctly. It's so small and it's so soft. We even looked for very good surgeons for microsurgery, and we never really managed to do it. So on that slide, you see one of the very few experiments where the staining went inside the tube, so we know it's luminized, but we always break it. You see here where my pointer is, we cannot sustain that perfusion for long. So now we are trying to anastomose these vessels with the capillary beds that Bruna is building to see if we can connect and perfuse them in a different way. But to detach them and analyze with PCR or a lot of different tools, yes, we are able to do it.
Very nice. Do you think if you put some parenchyma cells, I'm thinking about hepatocytes maybe, in the dome, in the long period, you can have like a kind of organoid surrounding the new vessel?
Yeah, that's another thing we would like to do. And we haven't used hepatocytes yet because we don't use cell lines. We use patient cells, and they're, as you may think and know, it's you want to be very delicate with them, and you want to use them in good experiments, And so we haven't tested yet, but Wenjin, the researcher of my team, will be doing that. And what she has done already is the co-culturing with non-parenchymal cells, but there are still neighboring cells in the liver. You remember the ones that I showed you, the hepatic stellate cells? And they're usually activated in culture with the L-sex. But we found that when you co-culture them around the vessels, if you place them when the vessels are being formed, they stay round, they show no alpha smooth muscle actin, so no activation. And they even seem to be helping new vessels to be formed. So this is very preliminary. This is from a month ago, and she's still working on that. But the co-culturing is definitely getting better than when you have the L-sex as monolayers. So your question is very good. And again, all the cues and the key markers of L-sex are present for longer than when we don't have the vessel. So we think we're improving the model. And of course, there's a lot to be made still. Thanks for your question.
Thank you, Matthew. Maybe the last one, have you analyzed the forces that are being deployed over the dome when those videos, when you show that cells are pulling? Have you analyzed the forces that are made inside this dome? Yes, these images.
Yeah. Again, a very good question. We haven't, for a simple reason that this is 3D, and we're talking almost the vertical edge of the dome. I know how to interpret, it's called traction force microscopy. I know how to interpret forces in a gel when I know on which plane I am. If beads are going all over the place in 3D, I have no idea how it works. And we talked to the experts. Actually, we did this experiment in Benoît Ladoux and René-Marc Meige's lab, who are the world experts in that. And we were only allowed to talk about displacement of beads. So the arrows you see are just displacement over time, because that's what we can measure. But the forces, I think now we are not able to measure them. A paper was published a month ago by Xavier Trepat's team in Barcelona, which is the first paper which you can actually trust they're measuring forces in 3D inside soft gels. I haven't read it yet. I have no time at the moment, but I think it's very, very tricky. But that's where we want to go. Maybe we go for thinner gels. We'll see. But at the moment, we don't know. I'm making only long answers to your short questions. I'm sorry.
Aquí tenemos otra pregunta de Janu.
Adelante.
Hi, Matthew. Just out of curiosity. So if I understood correctly, for these chips that you have, like the gel matrix, where you were growing the vessels, like embedded in the gel, you said you needed soft gels for them to grow properly, right? So I was just curious, you know, if you have worked with like more rigid gels and what happens with them.
Yeah, so for those cells to actually form, you see here on day 3, they only start forming the matrix. If they are too stiff, cells will not be able to deform them. If I go back to this paper here, they have shown that the stiffness is very important for those chains of force to be formed by cells. So you have how can I say that? It's like a collaborative effect between the cell's contractility and the amount of deformation you can apply to the gel. So if it's too stiff, it won't work. And actually, the question, if I can go beyond what you just asked, we were playing a lot with collagen gels, which are good for this type of self-organized structures. And we switched to fibrin recently, because I don't know if Milica told you about that, but fibrin is good for temporal gel. So cells are feeling like it's soft enough to organize. But if you add the fibroblast, they will completely replace the fibrin, fibrinogen plus thrombin structure by a new ECM that's more prone to be tissue specific. So if you use too stiff gels, this won't happen. And I'm also sure that the interstitial flow won't be as good as you would expect. So I'm not sure this will work. But the density of the concentration of the gel you use and the density of cell are very tricky to control to make this happen.
You say that it won't work with. Sorry. When you say that it won't work with the rigid gels, you mean that the cells just stay dispersed and they don't form not any self-organized structure, or do they form some other self-organized structure that are not vasculature structures?
No, they don't form anything. They are not able to pull on the substrate anymore. And even if you use high density, actually, we had that discussion with Bruna yesterday. She had some very weird results, and she couldn't make any. They couldn't form any structure, and she realized that the density of cells was too low. She miscalculated, and that can happen. And just that, even in a soft gel, they were staying round-ish where they were actually placed, and they were dying. So if they don't form anything and they stay on their own, even if they have medium and everything that they need, it's not like in 2D. It's like they need their neighbors, if you want. So if they don't form any structure, they die. And we haven't tried very stiff, but the stiffer gel will make no structure and they die.
In the streaming, we have one question.
Yeah.
Aime Torres say, hello, Matthew. Thank you for your great presentation. I have a question. Now you guys have a lumen. Is the next step to try to impose flow?
Yeah, that's exactly where we are today. It's been a long time now. Actually, we are kind of frustrated that we cannot perfuse those vessels because they're way too soft. So when you have them embedded, it's easier because they're connected to the side. So it's easier to perfuse. They're actually perfusable thanks to this interstitial flow you made. And now when we try to poke at them, when we try to interconnect them with microfluidic channels, they don't open to the channel. So that's our goal, to perfuse. And as I was saying earlier, I don't know if you heard the answer, but now our approach is to do, I will go back to this slide. Imagine the spheroid that you see here is not a spheroid, but is the vessel that I showed that we can make. We would like to interface, like here at the top of the chip, we will place the vessel that we can cut and place here. And we hope that we can anastomose those two by interstitial flow and growth factor gradients, which is what we are starting in January. We recruited a student to start on that.
Okay.
I'm giving out all my ideas, but it's good. If you can do it before us, let us know.
Well, thank you a lot, Matthew. See you soon.
No, thank you. Yeah.
So we are in contact. Now, I present Dr. Akeya Gupta. She's researcher. She's researcher in CNRS, is the equivalent at the CONACYT, but in France. She worked in the, bueno, mejor en español. Desarrolla su trabajo en el Centro Interdisciplinario de Nanociencia en Marsella y ha implementado novedosas herramientas ópticas y de nanoestructuración de superficies para investigar la adhesión y las propiedades mecánicas de las células T en particular y sistemas biomiméticos. Además, colabora internacionalmente en proyectos interdisciplinarios que combinan física, química y biología. Ha trabajado en temas fundamentales que abarcan la biofísica de membranas, la mecánica celular y la interacción entre sistemas biológicos y nanoestructurados. Dentro de sus áreas principales de investigación está la adhesión y organización de membranas celulares, también técnicas ópticas y de nanofabricación, así como herramientas experimentales para medición de fuerzas de atracción. Entonces, ahorita ella nos presentará su trabajo. Creo que es el conector más bien.
So, thank you very much for the introduction. Thank you very much for the introduction, and thank you for the invitation. I put here some things that I really liked about the visit in addition to science. So this is the last talk, and I see that we are not very many. So what I'm going to do is to request all of you to please come forward, because I want the talk to be interactive. I want you to stop me if you don't understand something and ask the questions right away. Please. And in fact, I prepared a challenge, okay? There is a little prize for the best student question. I don't know how many students are still there in the room. Are there any master students or younger in the room? No? Yes, one? Is there any PhD student in this room? Damn. Luckily, I put postdoc, and I think there's at least one postdoc. Well, you're still a PhD student. Are you the only one? Are there any postdocs in the room? No. Okay, so if you ask a question, then you get the prize, right? And Lauro and Pieroni will judge whether this is. Okay, I think now you don't need the microphone. Can you hear me? Behind, yes? Can you hear me, please? Yes? You can't hear? No? Can you? Yes? Yes, but if you're here, d'accord. Okay. Is this okay? Yeah? Perfect. Okay. All right. So thank you very much for coming forward. And thank you very much for stopping me, like in advance. I'm thanking you for stopping me and you don't understand something. I don't have to go to the end of the talk. More important that we interact. All right? I will start with a small tribute to my mentor, Professor Eric Sackman. Is this good? Yeah? Thank you. Who was a mentor to me and also to Laurent. He passed away, unfortunately, this year. He was the founder of biological physics in Germany. He's considered to be the founder of biological physics in Germany, and he's among the pioneers for let's say biophysics worldwide. He was an enthusiastic and dedicated teacher, inspiring and caring mentor. And from him, I learned that, in research, you need intuition, but you also need rigor. So these are the two books he wrote about during his lifetime. So this one is called Handbook of Membranes. And if there is any membrane person in this room, you must have come across articles from this book, because this is really the fundamentals, even today. This was published in the 80s, I think. And throughout his lifetime of lecture, he wrote towards the end of his life, after retirement, this textbook, which is unfortunately still only in German, but English translation should come out. And I put these cartoons because, whenever you went to see him, he would take out a pencil and crayons and start drawing these cartoons and inspire us from that. If you see cartoons like that, very possibly these dead bats too. There's some link with Professor Sackman. Okay, so now to the topic. I'm going to talk about, so the T will, for the moment, stay aside. When you talk about cells in their microenvironment. So in their environment, micro, nano, whatever. And so if I want to talk about how cells interact with their environment, so I took this logo to mean a cell. I hope this was correct. There's a nucleus, there's a membrane, so this is a cell. To interact with its environment, it has to adhere, right? So adhesion. Now let's consider first its interaction with another cell, which is part of the environment, right? So there are two cells, they need to meet, and they need to form bonds to hold them together. They need to deform and form an interface through which they will interact, okay? The first part of my talk, I'm going to talk about only artificial cells. Why? Because you see already in this drawing, in this cartoon, how complicated a cell is, all right? So to do really physics from fundamentals, I'm going to simplify the cell. Okay, let's go back many years when people first started wondering how cells adhere. You see the date, this is 1961. People didn't even know what the cell membrane actually looks like. So the most known model of cell membrane, which we more or less accept even today, the fluid mosaic model, Singer and Nicholson, this paper came out in, no? The year I was born, which is 1972, long time ago. But this is even earlier. People had no idea. Integrins were not discovered. So they thought, this is S.G. Curtis, who was also a pioneer in cell biophysics, Englishman. Okay, these things are all like messed up. This is not what I wanted to point out. What I want to point out is that he thought that electrostatic potential, van der Waals forces, and the deformability of the surface, this is what keeps cells together, okay? Now we know that he was very wrong because he didn't know about proteins. Now we know that there are glues, proteins are the glues which glue cells together. But the point I'm going to make is that physical interactions, electrostatic, van der Waals, etc., are still important, right? So the cell has to live with it. Physics is always there. You can add biology on top, but you can't get rid of physics. I forgot who started. I think this was Danielle who was making the point about, you were making the point, right. Okay. So with this idea, very early experiments, they took, this you can think of as a test cell. It's a liposome, it's a giant unilamellar vesicle. Basically, you take a cell, you take out everything, and you just have the membrane, okay? It's like a balloon of made of lipids. Inside your water, outside your water, okay? You hold it in a micropipette, you pull slightly, and you see, you'll get it interact with the surface or with another cell, You look at the deformation. So before interaction, this is kind of round. After deformation, it's deformed. From the shape change, you can calculate the addition energy. So you see that this is kind of complicated because you need to pull, you need to, and then you need to look at the shape, and this is very limiting. So there's another way to do it, which is this approach that I learned from Professor Sackman, which is that you prepare your surface and you let your, whatever it is, the cell membrane or membrane of your liposome interact with the surface. And then you map the adhesion. You just let it do what it does. You map the adhesion. You see how the distance between this membrane and the surface is changing, okay? This is a very, very generic way to approach a problem in physics. If you think of Newton, to make his laws, what did he do? He looked at how the distance from the sun to the planets are changing, right? So to measure distance between two bodies is a very fundamental way to look at their interactions, and that's what we're going to do. The question is, how do I measure this distance? Okay, this distance is very, very small. It's of the order of a few hundreds of nanometers, maybe, around. So what is the best way to measure small distances? Who did optics lessons in this room? Exactly, interference. Exactly. So interference is a technique which is known for a very long time, okay? This is the reason, because of interference, that you see colors in a soap bubble, yeah? When you're young, colors, right? Or a bit of oil in water at the side of the road, you see colors. 1664. Many, many years later, in 1964, this was, Brownian motion. Curtis again, about who I talked about just before, he had the idea of using interference to look at cell adhesion. But you see, he had very ugly pictures. This was very difficult to quantify. And people thought this was a great idea. They tried fairly hard for 10 years, 15 years, and they gave up. Eric Sackman had a different idea. He said that, okay, let's forget cells. Let's use this technique to look at objects which are simpler optically. So beads, or we will see also liposomes. So this is just a bead, yeah? Just, how do you say bead in Spanish? Okay, so that, right? And if you did basic physics practicals, maybe you came across Newton's rings, yeah? So if you do interference with the spherical objects, then you expect fringes like this, yeah, rings. By looking at the intensity at the center, you can measure the distance between the bead and the surface. Because the intensity, this is the intensity axis, this is the height, so height of the bead above this surface. This goes as a sinusoid, all right? And that's why you have fringes, because when it's very close to the surface, you see dark. If it goes further away, you see white, et cetera, et cetera. And the shape, like, as the surface is going away, the shape is following the fringes. So that's how you can reconstruct the shape. And with nanometric precision for simple objects like beads, and also for vesicles. So this is a giant unilamellar vesicle. This is the membrane. Okay, this is going to be my system for the first part of my talk. And what I'm interested in is not what's happening at the top, but just simply what is the distance between this membrane and the substrate. So in an experiment, it looks like this. Okay. What I'm seeing is that the membrane has interacted with the surface. It's kind of flat. At the edge, this is going up. So it's like if you put a bowl on a glass table, you go under the table and you look up. Okay, so the edge of the bowl is going away from you. That's why you have these fringes, which I'm going to ignore for the moment. but there is fluctuation here. Okay, so the membrane is fluctuating. These fluctuations are thermal fluctuations. I think fluctuation is the word in more or less every European language, right? You understand what I mean by fluctuation. Okay, so why does the membrane thermally fluctuate? So what is thermal fluctuation? Thermal fluctuation is simply that because of ambient temperature, okay, if you have something which is light enough, like which can move easily enough, then simply because of temperature, it's going to move. It's like Brownian motion. You all know about Brownian motion. In a sunny day, if you are on a dusty street, you can see this, the dust particles moving, right? This is in a room when you're cleaning, rays come in, you see particles going in and out. So this is, the same phenomenon. This membrane is so soft that just because the water particles, they're moving because of temperature, they hit the membrane and they make it move, okay? All right. Now, from that image, I need to reconstruct the height, okay? So I'm going to look at the intensity. If I'm sure that I'm close enough to the surface, if I'm on this branch, then I can simply read off the height. If I'm not sure, what I can do is I can add another color. So if I, instead of looking in green, I look in blue together with the green. So then this form is different. And then you have, with the two information, you have broken the degeneracy, and you really know where you are. Then there's another subtlety, that if you're using a liposome, it's not the same as beads. You don't have a sinusoid which starts from 0, but it starts there. Okay, I'm not going to go into huge detail, but if anyone is interested in doing RICM, please feel free to contact me or Laurent, and we are very happy to teach you. Now, you have been seeing I've been saying fluctuation, fluctuation, fluctuation. We have to measure fluctuations. The thing to remember is that the intensity fluctuation, okay, if I look at for the same height fluctuation, depending on whether the intensity is here or here, you can have very different, like whether the membrane is here or here, you can have very different intensity change. Why? You see here the X is same, but the Y is enormous because I'm sitting at a place where this is linear. Here I'm close to maxima or minima. For the same change in height, I will have very little change in intensity. So you have to be careful about all this, all right? So if I do all this very carefully, then what I can do is, so here you have a liposome. So this is the cartoon. It's sitting on a stamped microgrid. I think you saw this many, many times. And I'm looking at this part of the vesicle. We do this simply to give a nice shape to it. It becomes easier to handle things instead of just, the image that you saw before. So then it forms pockets, and then I can look at this, reconstruct this frame by frame, look at each time point where this membrane, what it looks like, and we can calculate the interaction potential between the membrane and the surface. So the membrane is fluctuating a little bit, and it's around 100 nanometer away. Now we have to look at adhesion. Okay. Okay. Let me see what. Okay, let me introduce this idea already. Now I have to look at adhesion. This membrane has to adhere, right? It's 100 nanometer away. It's going to use a protein to adhere. What's the size of a protein, roughly? 10 nanometer. 10 nanometer, exactly. So, my protein is here, and I have to shake hands with you, right? 10 times further away, 10 meters away. That's why fluctuation is very important, because how am I going to hold somebody's hand there? I have to move. I'm connected to the whole membrane, so the whole membrane is moving. All right, so that's how they make contact. Okay, now, if the interaction between the surface and the membrane is strong, And maybe, maybe it's, okay, electrostatic is no good underwater because it's damped, but still, okay. If it has some kind of very strong interaction, including with the help of proteins, this is what it looks like, okay, after addition. You may have fringes, you may have very strong addition, you don't even see fringes, and this is balance of addition and elastic energy, okay? And the dynamics is dominated by hydrodynamics, meaning when the membrane is adhering, it has to push away the water, and this is what is determining the time scale. This was studied many years ago, 30 years ago, very boring. We didn't want to go into that because it was already known. What we wanted to look at is the limit where the addition is very weak, okay? Weak addition, then along with enthalpy, which is like the bond energy, something else becomes important, which is entropy. Everyone knows what is entropy? Someone doesn't know what is entropy? What is entropy? Disorder? We'll take that, disorder. So the second law of thermodynamics says that a given system tries to lower its free energy, so by maximizing its entropy. Okay, it's kind of equivalent. So when the energy gain by forming bonds is very little, then entropy can become very important. Okay, so some bonds will say that, okay, I don't want to adhere because then I have the freedom to be disordered, because addition, you get stuck, it's kind of, you're imposing a kind of order. Okay, and in this case, the dynamics is determined by how fast these molecules can react when they come together, reaction, and how fast they find each other, diffusion, okay? All right, so can someone give me example of a ligand receptor which is weak? In fact, all biological bonds are weak. Integrins, biologists will say it's a very strong bond. Physicists comes. 7 kT, what is kT? K is the Boltzmann constant, T is the temperature. If you multiply them, this is a unit of energy, and 1 kT is like the energy that you can pull out of air. Why? Because if you remember your statistical mechanics, there is like equipartition of energy, and then, one hit is roughly 1 kT. Right, so kT is the energy we pull out of thin air. 7 times kT, if it happens that 7 molecules push together, okay, the bond breaks. Only 7. Cadherin, which holds our tissue together, right? All of you know that. It's only of the order of 2 or 3 kT. Very, very, very weak. So I'm going to look at cadherin addition. And you will see that why the tissues are held together. This is because of cooperativity, because you can stabilize things. All right. So this is my system. These are cadherin molecules. This is like the adhesion patch which is forming. This is simulation. This is RICM that you saw, that I explained before. You saw it in Laurent's talk. Daniel talked about it. Pierre talked about it a bit. So basically, when you see dark, this is adhering. When you see white, this is less adhering in general. So what we did was, because we wanted to look at the role of fluctuations, we changed the tension of the membrane. So when the membrane is tense, it tends to fluctuate less. So the membranes we are used to in everyday life, right? The membrane of a balloon or even soap bubbles, these are very, very stiff. They don't fluctuate thermally. But still you can imagine that if you blow a balloon very little, it's softer. If when you blow it a lot, it's more difficult to deform it. For liposomes, it's the same. So we deflate the liposome, then it fluctuates more. And depending on how much we deflated the liposome, we have this kind of zoo of behavior. Strong addition, is this even addition? Because this fluctuates a lot, and then these kind of things, okay? So I will not go into the details of how we did the analysis and so on, because it will take too long. What I want you to appreciate is that fluctuations has an enormous impact on the adhesion of this system. First of all, because of accessibility, like what I said, if the two membranes stay apart 100 nanometer away, don't fluctuate, they will never adhere, okay? For cell membranes, it's the same. For cell membranes, there is the glycocalyx, which is keeping the two cells apart. And if there was no means to penetrate the glycocalyx and make the first bond will never happen. And that's where what Pierre was talking about, tiptoe, that becomes important. We'll see this later. So accessibility. So you can say that, all right, fluctuations are going to make adhesion better because you can form more bonds. But there's another problem, which is that once you form a bond, let's say I've managed to form one bond, you heard again and again throughout this conference that biological bonds are force sensitive. So slip bonds, you pull on them, they become more fragile. So for bond which forms alone here, the membrane is still fluctuating. Remember, it's pulling on the bond. It's very fragile, it will break. But if you manage to form 3 or 4 together, then the middle one, this is protected from fluctuations. That stabilizes the bond, okay? So then you can form large bond clusters. You can keep the membrane adhered. So we use these ideas to do, here are the simulations from my colleague, Anna Smith. She's a professor in Germany. And these are the experiments, all right? And they kind of look similar. You have to increase fluctuation. So this is increasing fluctuation, and this is increasing heights, because obviously it also depends on how far you are to begin with. So when you have, let's say, very little fluctuation, and you are close to the, you're not very far either, you're little fluctuation, you manage to form bonds, and you make a strong adhesion. Okay, so now what happens is that, as you saw, domains are stabilized and single bonds break. So this gives the impression that there is a long-range attraction in the plane of the membrane. And this is interesting biologically because there was a long-standing question in the field whether cadherins can have lateral interactions in addition to the trans interactions, okay? So what we say is that, well, maybe there is some molecular mechanism at very, very short range, but at long range, this is simply membrane phenomenon which is bringing them together, okay? Okay, how much time do I have? I forgot to set my timer. 15 minutes, all right. Okay, so the next part of my talk is about T cells. In the microenvironment, Pierre already gave a nice introduction. So, T cell, target cell, the system is this. Here I write 20 nanometer. We now know that it's maybe before addition, which is more like 100 nanometer. Then they have to form a bond. So I'm going to either use only an antibody against the TCR or antibody against the TCR as well as against the integrin. Very similar to the system that Pierre talked about. And I just want you to notice that this interface, I'm going to look at this interface and how it forms. And this is very dynamic. My approach is to replace the antigen presenting cell with which the T cell was interacting with the antigen presenting surface. This is, again, something that Pierre, J. Groves, Laurent, everyone introduced. And then what we do in the lab is to vary some property on the surface, which can be ligand mobility, which can be stiffness, which I will not talk much about because Pierre talked about it, and clustering. This will be what I talk to you about mainly for the next 15 minutes. The strategy is to image the interface, and the goal is to understand how to bridge molecular and cellular landscapes. So this is echoing what Pierre said. These are the collaborators. So Laurent and Pierre are the main collaborators for this. Okay, very quickly. I said that for beads and liposomes, we can do RICM with 1 nanometer resolution. Cells are much more complicated. I'm not going to do quantification of cell, cell to surface distance today, but we can do it. If anyone is interested, we can talk further later. Today, I'm going to use RICM just to segment the spread area like Laurence showed and Pierre showed. So here, these cells are on mobile or immobile surfaces. And you see that on supported lipid bilayer, when these things are stuck. This is on supported lipid bilayer, which we made in a way that these guys don't move anymore, the anchors, right? Then the cell spreads monotonically. There is nice retrograde actin flow, which is a classical thing. The conditions that Jay Grove showed, this is what you will see. But he had mobile ligands, but he has integrin in addition to ligand for TCR. But if you don't have integrins, what happens is that the cell tries to spread, but it can't, all right? If you add integrins, then you rescue spreading. And then here, I don't know if you guys know a little bit about immune synapse, you will see that this is the classical synapse. Okay, so we did many experiments like this with different kinds of drugs on different kinds of surfaces, and I tell you the final story. The final story on homogeneous surface is this, that the cell can access only limited number of morphologies. So time is increasing like this, cell comes down, it starts to interact with the surface. I'm looking at the actin, this is labeled. So it forms a first contact, which is formin dependent, and then it goes into an up to 3 dependent phase. So formins help cells form finger-like structures, right? Like this. And up to 3 helps it to form lamellipodia, structures which are flat and which spread. If there is friction, meaning that the linkers are stuck, then the cell is able to spread. If the linkers are mobile, then the cells are not able to spread. If I put integrin, the cells are always able to spread, stuck or not. So all this, and on soft, as Pierre showed, we are always in this regime without integrins and in this regime with integrins. I think I will skip this because Pierre explained this model. We can understand cell spreading with the help of this model where there is an actin pushing on the edge of the cell and helping the cell to spread. Okay, so now the point is that it's known, factors which determine cell fate or morphology, there are different factors, okay? Chemical is very well known. Obviously, you put some toxin, it's going to die, right? It needs food, etc. Mechanical is something that we have been talking about. I'm going to talk about now of spatial patterning, like geometry, which can be either topography or chemical. I'm going to talk about the chemistry. And this is a quote from Wolfgang Pauli, who said, God made the bulk, surfaces were invented by the devil. And I think Matthew made a comment on that, that surfaces, whoever worked with that, you know that it's horrible. No, I think it was Pierre who said that you can have the protocol, but it's not going to work for surfaces. All right. So all this story of patterning goes back to this paper. Okay, in 97, Ingber's group, they stamped fibronectin patches, and they showed that cells take the form of these patches. But what surprised them was that if you don't let them spread, these two, they just die. If you confine the cells, they die. So this was the first time that we knew, number one, that we could pattern the cells with stamping proteins, and number two, that their even survival depends on how well they can spread, how well they can interact. And now, I don't have to tell this to this audience, you can pretty much make the cells do what you like by stamping the right way. But all this is micron scale, okay? How about if we go even smaller and go to nanoscale? So proteins are typically 5 to 10, maybe 70 nanometer, not more. So does the cell even care if I have proteins thrown on the surface randomly or in some kind of pattern? So these are experiments from the lab of Joachim Spatz, and he showed that if you have, depending on the spacing of these are gold nanodots on which there is one RGD molecule on each dot, and in between there is some repulsive molecules. Depending on the spacing, the cells may be able to spread normally, form the actin fibers, etc. Or if the spacing is too big, then they're not able to do that. And they have some clever means to show that it's really the spacing that matters and not the density. So if you take the same density, but some of the molecules are closer, then the cell is still able to spread. They concluded, they concluded, not them actually, later papers concluded that this spacing comes from the size of tailid. So if you're forming the focal adhesion, first there's the integrin which binds to the fibronectin, then the integrin has to cross-link. And this cross-linking protein, I forgot if it's tailin or vinculin, which one is giving the scaling, but one of those, this is unable to therefore form focal adhesion. So when I started many years ago, my question was, okay, T cells, they don't form focal adhesion. They form just, adhesion patches which never mature to focal adhesion. So what about them? Do they have some kind of scaling? So then we spend a lot of time making micro to sub-micrometer patterns like this. One idea is to manipulate the placing of the ligands and therefore the receptors. You saw this similar kind of idea with geometric mutation. And the other point is that people think that the ligands of TCR, they themselves might be clustered on the antigen presenting cells. Okay? So my substrate looks like this. There's no metal. Why did I spend a lot of time making stuff without metal? The reason is that if I want to use advanced microscopy with high power laser, if I have metal nanodots, light can interact strongly with metal. So that's not good. So our patterns are metal free. I have some adhesive and activating spots and repulsive regions, all right? We can either do self-assembly or we can do E-beam lithography. I don't have time to talk about the techniques. I'm very happy to discuss afterwards if you want. We can vary the size. Now we are down to 40 nanometers. We didn't put cells on them yet, so I won't show you those patterns. And then cells, what happens? The TCR, it gets nicely, it makes nice patterns which reflect the underlying pattern, right? You see here this pattern, right? If you take a fast Fourier transform of the TCR arrangement, you even see these nice diffraction patterns. This is the RICM, so the adhesion is also patterned. Actin is sometimes patterned, so I'm going to come back to actin later. So we did these experiments, we measured the adhesion, and what did we see? We see that in case of T cells, they care about density. They don't care about the spacing. What they care about is density, okay? This is the take-home message from this slide. And since then, so we used spreading area as a proxy for activation. Since then, people have shown that really even activation, if you measure interleukin, et cetera, they depend on molecular density and not on spacing. That's a little bit puzzling because the pattern is actually having a very strong local effect. So strong local effect of the pattern, global effect, only density. However, now I have a system where I can put integrin outside of the dots, okay? I do that, and then I compare at the same density, homogeneous and pattern, okay? Let me see. So this is homogeneous, this is patterned, this is the RICM on homogeneous, and these are two kinds of behavior we see on the pattern. The pattern is the same, and the same substrate, sometimes they form nice, visible structures, sometimes not. But what is interesting is that globally cells spread more when they are on pattern, meaning that now in presence of integrin, even the global property depends on patterning. Okay, sorry, here, okay? Now, the TCR is still nicely clustered. And if I look at this kinase ZAP70, which comes on top of the TCR, this is the first step of activation. So this kinase is also nicely structured, okay? And it doesn't matter whether there is ICAM or not. Okay, so the take-home message is that there is a dual scale of T-cell response. So when the ligand for TCR complex is alone, then the cell does not care about the nanopattern. It cares about global averages for global properties. When there is integrin, then both local and global properties depend on the nanopattern. So cell scale TCR clustering matters only if integrins are also present. So I will stop very soon after this, okay? I just can't resist showing you this experiment where there's what you see is actin, okay? Cell is coming down, making contact with a pattern substrate. And what you're going to see is that the actin lights up where there is pattern, okay? The actin is first nicely co-localizing with the patterns, and then this co-localization disappears. So I'm just showing you a bunch of observation now, okay? We don't really know the mechanism of why this happens. We quantified the final state with the help of some fairly involved calculations. We can talk about this later. But the clustering TCR has a very subtle impact on actin architecture. And now we are doing super resolution of the actin to know, we think that maybe the patterns are acting as some kind of nodes for the actin. Okay. So to sum up, to bridge scales, molecules are clustering on the membrane and transmitting this message to cell scale. So mesoscale membrane and actin cytoskeleton may be key players in this bridging of scales. So I'm almost finished with my talk. I wanted to put this slide. This physicist, she reaches heaven and asks God, what is the meaning of life? And God replies, do you mean in vitro or in vivo? So then she says, well, the experiments I did in vitro should follow the same laws as what happens in vivo. And God says, great, you have achieved nirvana, you can enter heaven. Okay, so I wanted to thank all the people with who I've worked over the years. I think I already thanked the specific people whose work I presented. I wanted to say that, this is where I work. I'm in this building, Pierre-Henri and Long are not very far. There are different kind of funding opportunities for short visits and internships. I think this network can support. For other kinds of grants, for PhD and postdoc, there are other kinds of grants. So if whatever stage of career you're in, if you want to visit us, work with us, contact us. If you're students who want to do that, advertisement, okay? And now we are back to the challenge and open to questions. Thank you.
we need to use the micro? Or
People are close enough. Some people came later and sat behind. Could you hear me?
yeah.
Well.
Okay. Someone question? Yeah.
So please feel free to also ask in Spanish if you're more comfortable. Pierre-Henri kindly agreed to translate. And so that I'm not biased, I asked Pierre-Henri and Laurent to be judge of who gets the prize.
Thank you so much. I'm start thinking about the materials in which these patterning pieces are made. What material are them?
Okay, so you're asking about the final pattern, right?
Yeah.
What
yeah.
does it? So this is glass.
Okay.
On which, wherever there is a patch, I first have a layer of organoaminosilan, on which there is BSA biotin, on which there is neutrovidin, on which there is the biotin-related antibody.
Okay.
Outside, this is PLL PEG, or supported lipid bilayer. So polylysine PEG, like polyethylene glycol. So this is basically very standard way to passivate glass. You could also use, what is it called? Fluoronic acid, but not directly on glass. Then you have to, so we have tried different chemistry. It can be fibronectin. I didn't have time to show this. I think I have a slide on that. So that's actually a nice question because right. So this is a very different chemistry where we have fibronectin, and then these are fibroblast cells, and you see they are also nicely patterned. These are 700 nanometer patterns, but I guess if you go down, we should be able to go down.
I was thinking about that because of maybe, well, do you know how biocompatible can be this arrange that you are describing me? How biocompatible maybe inside a body can be this arrange?
Inside the body.
Yeah, yeah.
Obviously, we will not put glass inside the body.
Oh, yeah, for sure.
But so that's, we have to think. So there are collaborators who asked, can you do this on colloidal beads? Not with the technique that we developed, but definitely this can be extended. We can do this on PDMS, but flat. I haven't tried with, because in the body, if you're thinking of injection, this is, I think it has to be a particle. If you're thinking of implants, I think this is very feasible to do it on titanium oxide or plastic or whatever.
It's because the final part of what I was thinking is that seeing that the immune system can be activated by this kind of patterning, some of the problem with cancer is that it hides, tumor hides
Absolutely.
from the immune system. And in some cases, maybe an implant of some material patterned this way, maybe not the same materials, but patterned with the same shapes, can activate and can enforce the immune system to attack these tumors. So I was thinking about that.
Yeah, why not? It's a nice idea.
Thanks so much.
Aha, now there are many students, but you missed the talk. Did you hear the talk online? Maybe they can ask questions, because otherwise this guy gets the prize.
Hi, Kenya.
Hi.
Thank you. My question is, I think that many of us have been more familiar with changing the substrate stiffness, like to induce changes or differentiation in the cells. Do you think there is a scale relation between the distribution of your patterns and the stiffness of the substrate?
That's a very good question. I would love to do these experiments. We now know how to stamp pattern on soft substrate, but we haven't, done systematically putting our cells on it. I don't want to, anticipate, because T cells are so unexpected, because what Pianori showed the behavior that all other cells on soft, if you make the substrate stiffer and stiffer, for all other cells, they spread more and more, and then they saturate. Then, for T cells, they will spread more and more, but then there is a peak, and then they spread less and less. I think so, but I don't know the answer. I have to do the experiments. Thank you.
Another one question. Then I want to ask something. In the interface between the cell and the substrate, it's very thin layer. But if the cell produce some molecule, it's possible, or do you have some, do you see something about the concentration of that molecule diffuse between in that interface? If it's possible in the size of that gap, see or identify a certain gradient of that molecule?
That's a very good question. So it really depends on what molecule, okay? So we did very clean experiments with liposomes where we either used biotin and avidin, or actually neutravidin, or we used cadherins. So if I look at the footprint of neutravidin, which is like avidin, right? So this is around 4 nanometer. The cadherins, these are not real cadherins that we use, but the cadherin extracellular domains, which were connected to only one lipid. So the footprint is much smaller. Now, what happens in the first system, where this looks large on the membrane, the system gets jammed, okay? Molecules diffuse in, they form a ring around the contact zone, and then no more molecules can come in. You increase the temperature a little bit, this fluidifies, okay? For cadherins, this does not happen because the footprint of our extracellular domain cadherin constructs, the footprint is very small. This is able to diffuse even at kind of high density. There is no traffic jam, and this diffusion can happen. So it really depends on the size of the protein you're looking at, and their interaction. If they're interacting strongly with each other, forming kind of jam system or some other kind of structure.
Okay. Thank you. Thank you a lot. Someone else?
Thank you, Kaya, for your nice talk. I have a question. I think that, I don't know if it is difficult. When the cell has his better function, when it's all spread or when it's a little bit spread on the surface? How can we measure when the cells are acting like a good cell?
Right, so we are talking about T cells, right? So T cells are good when they're activated correctly, right? So if they see the right molecules, they should be activated. Now, a long time ago, Pierre Bongrand, who was the founder of the lab of Laurent and Pianori, he showed in experiments that there is a correlation between the spreading of the T cells, the surfaces on which they spread well. These are also surfaces in which they get better activated, more interleukin secretion, and more proliferation. In general, this is true on glass, but on soft or on pattern, this can be very different. So we have not really looked at a lot on activation, on soften pattern, but what we did was to look at activation on mobile and immobile. And this is very interesting because on mobile, the cells are not able to spread, but they are very well able to activate. So this is a very good question, and we need to understand this connection between spreading and activation better.
Thank you so much for the talk. I really like this topic. When I was in the PhD, I was studying something about the cell adhesions. But I have a question. I was wondering about the soft substrated also when is elastic, but when the mechanics properties of the substrate are viscoelastic, modified the spreading, also the velocity of the spreading. Sometimes in soft substrate, you can observe the spreading of the cells. I don't know if this happened with T cells also, because in the monoliths have many difference when the with the adhesion cell. So I don't
Sorry,
know.
which one? Which cell did you talk about?
For example, the epithelial cells are adhesion cells. That is the nature of that cells. But the T cells is not. The T cell is in the blood. So I don't know if the viscoelasticity of the substrate modify the spreading of the cell.
Yeah, so I did not show this data, but this is something that we worked on with Piori and Luo. Tissue forming cells, yes, indeed, tissue forming cells, they actually, most of them, okay, not all of them, but most of them spread more on stiff, less on soft, even if soft is more physiological, but they spread more on stiff. T cells, they have an optimal stiffness on which they spread the best. So very soft, they are not able to spread. Around 20 kilopascal, they spread very nicely. But if you make it harder and harder, they spread less and less. Is that the question?
Yeah, this explains something, but I mean, for instance, when the viscoelasticity have two parts. One is the elastic part, and the other is the viscous one. When the elastic part is slow, like 2 kilopascals,
yes,
but they increase the viscous one, you can observe spreading.
Oh, I see. I see.
I mean,
but how will you keep the stiffness same and you introduce viscosity instead of this being purely elastic? Is that what you're saying?
Yes. For example, in a polyacrylamide gel, you introduce a linear change for increase the viscoelastic, the viscosity of the gel. So some experiments shows that you can observe spreading of the in soft.
So I think I need to discuss better with you, because if you don't have viscosity in a purely elastic substrate, right, you can imagine that the viscosity is in fact infinite, right? So if you're introducing viscosity, what you're doing is reducing the viscosity, not increasing it, right? Correct? Because what is viscosity? Viscosity is the resistance to flow, right?
Yeah.
So if you have a solid, it's going to resist flow. It's never going to flow. So it's infinitely viscous. Now you make it less viscous by, even if it's high viscosity, you're actually making it less viscous by introducing viscosity. Is that what you're saying? That compared to elastic, a viscous, they spread better? Okay, I was not aware. We can discuss later.
Later. Thank you so much.
You're welcome.
Thank you. Well, this is the last talk. So we invite you to take a photo, please.
Before that, maybe Pierre has to say who won the prize.
Oh, good.
Or Laurent.
We want a prize, so. Pues, ese momento.
Okay, so I think the winner of the challenge will be you, even if I don't know your name. What is your name? Yuli. So your winner is Yuli.
Gracias. Las palabras al ganadora.
Thank you so much for coming to Mexico. It was a wonderful experience to have you here, sharing your knowledge, all the experience that you have in all this time. And I think that is very important that you come to Mexico, because many students need to know what is doing in other countries, and that they know that they can do some projects in other countries. So thank you so much for being here.
Thank you, and thank you for the invitation.
Bueno, entonces, si pueden pasar aquí enfrente, Gus nos va a tomar una foto, entonces a todos los asistentes. Yo no quería que saliera, pero bueno. Entonces, pásenle, por favor. ¿Hay forma de poner en la pantalla otra vez el logo? O lo photoshopea Gus ahí.